Hello. Hello, am I audible? Sunil sir, am I audible?
Hello.
Hello.
Yes, ma'am. Good afternoon.
Good afternoon, ma'am. Please wait for a minute. We are about to start. Just 1 minute, ma'am.
There are bits of information. I would like to make some announcements. Dear participants, those who are having their presentation in 2D, they are in this main session. You don't have to go anywhere. You don't have to join any breakout room. Once again, those who have their presentation in 2D through the schedule that has been posted to you, they will remain in this session. Those people who have their presentations in 2E and 2F, they are requested to join session 2B and 2C. So in the morning it was ABC and post-lunch which is DEF, but D remains in A, which is the main session. Those who are in B, they will remain in the session. Those who have their presentation in E, they will remain in 2B. So it's like you are matching B with E and you are matching C with F. So those who have their presentation in 2E will join 2B. Those who have their presentations into F will join 2C. Once again, those who have their presentation in 2D will remain here. Those who have their presentations in 2E will go to room 2B. Those who have their presentation in 2F will go to room 2C. Thank you so much. Now we are going to start our session 2D, which is also 2A. A very good afternoon to all. Thank you so much for joining the session. And I take this opportunity to introduce our session chair. And we have Dr. Anji Reddy Polu, who will be chairing the session. Dr. Anji Polu Reddy has completed his PhD from HS Gore Central University and postdoctoral studies from Sogang University, South Korea. He has more than 12 years of experience on battery materials and their applications in EVs. Dr. Reddy currently working as assistant professor in Department of Physics, BVRIT Hyderabad College of Engineering for Women, a premier engineering college in Hyderabad. Previously, Dr. Reddy worked in VIT University and KL University as assistant professor. He has published more than 50 papers in reputed journals and has been serving as a reviewer for many reputed journals like ACS, RSC, Elsevier, Wiley, Springer, etc. He has given many invited talks and keynote speeches in various countries and reputed international conferences. He received many awards and honors for his contribution in research on energy storage devices and also in academics. Dr. Reddy is having 3 patents to his credit. He has an H index of 22 and has more than 1435 citations. Dr. Reddy is also a life member in Electrochemical Society of India and has visited 12 countries. With that brief introduction, I invite Dr. Anji Reddy Kolu to session this chair. Sir, please. So kindly unmute yourself, Dr. Anji.
Yeah, right. Thank you. Thank you, Dr. Venus, for introducing me. Is it audible?
Yes, sir, it is audible.
Right. Thank you, Dr. Venus, for introducing me. I'm very happy to session this chair. In this session, we are having total 27 oral presentations, and I'm requesting everyone to follow the timings. The timing is 10 minutes. Out of 10 minutes, 8 minutes for presentation and 2 minutes for doubts, questions. Okay. I'm requesting everyone to follow the timing strictly. 8 minutes for presentation and 2 minutes for discussion. Okay. Now I am going to invite first presenter, uh, Rama Koyati. He is from India. Sir, can you share your screen? You can start your presentation. Rama Koyati. The first presentation, Rama Koyati. Yeah, you can start the. You can share your screen and start the presentation.
Dr. Rama, we can see you. Kindly start presenting.
Okay, ma'am. Hello, ma'am. Can you hear me?
Yeah.
Yes, ma'am.
Yeah. Good afternoon, sir. I'm Dr. Rama Koyati, Assistant Professor from Forensic Science Unit, Department of Chemistry, University College of Science, Osmania University. Today I'm going to present one of my paper entitled In Vitro Studies on Protein Binding of Polyhydroxybutyrate, which is isolated from Rhodosodomonas palustris KRPR02. So, coming to the introduction, there are a number of synthetic plastics which are sustainable in the environment for the long time, which are non-biodegradable, and because of accumulation of these plastics in the environment is producing a number of harmful effects on the living microorganisms and also showing a effect on the environment. So, in order to stop this particular problem, we have new approaches for the production of bioplastics, which is essential, which is used for the synthesis of your biopolymers using a biological method by using various biological sources like plant, animals, as well as microorganisms. Coming to the plants and animals requires a lot of time for their growth, whereas coming to the microorganisms, within a very short period of time, they can multiply in many more numbers and can produce various bioproducts. Therefore, in this particular study, I have selected the microorganisms as a biological source for the production of the biopolymers. And alkanolates are the biopolymers such as your PHB and your PHA, which are biodegradable, and for the production of this particular biopolymers also has very less cost and also eco-friendly in nature. And this biopolymers has many more applications in medicine, material science, as well as agriculture nowadays. The main aim and objectives of this is isolation of the anoxygenic phototrophic bacteria from the wastewater collected from the sugar industrial effluent, and after the isolation, identifying and characterization of the bacterial strains and utilization of this particular strains After checking for the production of the biopolymers and its applications in protein binding studies. So I have collected a sample from the sugar industrial effluent in Islamabad, and it was inoculated into the Bible and Fenning media, which is a general media, normal media. Samadar is sharing, ma'am. Hello.
Please wait. Yes.
So it was inoculated into the general media, which is called as a master sample. Later on, it was subcultured for many days, and it was grown in an enrichment media, and this was characterized for its microscopic morphological characters and phylogenetic analysis. Coming to this particular image, the first two pictures are of SEM images, which is showing the bacterial morphology, which is a rod-shaped bacteria, and also they're showing the size of the bacteria. And coming to the next two images is the TEM images, which are showing the internal structures, and also it is revealing the presence of the PHB granules inside the bacteria. And after the sequencing by 16S rRNA sequencing, and whenever it was submitted to the blast and identified that from the phylogenetic analysis, the bacteria that was isolated from this particular sugar industrial effluent is a novel strain of Rhodopseudomonas palustris. Later on, from this particular bacteria, I have extracted the PHB by using a hypochlorite and chloroform method. And the first image is the PHB that was extracted and purified by using a methanol and chloroform extraction. Later on, for the confirmation of the PHB, I have taken the sample and I have added the concentrated HCl solution. So here the biopolymer will be broken down into crotonic acid, and this a brown color solution is a confirmation of breaking down of the PHB into a simple crotonic acid. And this was confirmed by characterization by using a UV visible spectra here. So according to the literature, whatever the peak that we are getting confirms the presence of PHB. And here also you have a spectra of XRD and as well as the FTIR spectra, which is revealing the presence of PHB in the extracted sample. And here is a spectra of your NMR spectra of PHB extracted from the bacterial strain. So whenever we are comparing with the other papers, coming to the literature, we can confirm that whatever the sample that was extracted from the bacteria consists of PHB. So after the characterization of the PHB, here coming to the application part, here I have checked whether this particular extracted PHB has the capacity to bind with the protein or not. So this is a one experiment which was estimated by using a Lowry method and identified that as the concentration of the protein is increasing, the PHB binding capacity is also increasing. Coming to the conclusion of this particular paper is the intracellular PHB granules were extracted by using the hypochlorite and chloroform method, and the presence of PHB was identified by the chemical test by using a concentrated HCl solution, and the presence of PHB granules was also identified with the TEM analysis. And here the PHB extracted and identified by this bacteria were also confirmed by using instrumental analysis. Because of the binding efficiency of this protein, it can be used for the delivery of the proteins. And not only that, this particular polymers also used in the drug delivery. Not only this, coming to the future applications of biopolymers is if this particular biopolymer has an efficiency to bind with various products such as the nanocomposites and also certain drugs, which helps in delivering your various drugs into the body and also delivery of your nanocomposites into the agricultural field, which increases the yield of the crop. So not only the PHB extracted for the bacteria has an applications in drug delivery, but it also has an applications in agriculture, which the further studies need to be done. I acknowledge various departments, that is Department of Biochemistry, Usmania University, and Department of Chemistry, University College of Technology, Usmania University, and I also acknowledge the Central Facilities and Research Development, Usmania University, for providing me the necessary infrastructural facilities and their cooperation. Thank you. Any questions?
Yeah, thank you for your presentation, Dr. Rama. It's time for questions. We have time for one question.
Sir, your
Any
voice
questions?
is not audible, sir. Hello.
Is it audible now? Are you able to hear my voice?
Sir, we can hear you, but the volume is less.
Okay, okay. Are you able to hear now?
Hello.
This is
Hello?
the
Yeah, are you able to hear?
Yes, sir, we can hear you, sir.
Yeah, okay, okay. Thank you for your presentation, Dr. Rama. Okay. Now, if any questions, we have time for one question. If anyone have question, you can ask one question. If there is no questions, we can move to second presentation. Okay, no questions. Now we will move to second presentation. Yeah, the second presenter is Dr. Neeraj Shukla from India. You can start your presentation, sir. Dr. Neeraj Shukla from India.
Good afternoon, everyone. Is my screen visible?
Yeah, it's visible.
And I hope my voice is audible to everyone.
Yeah. Sorry.
Okay. Okay, so here I go. I have shared my screen and I'm going to start the presentation. So, okay, so first of all, I thank the organizers for giving me this opportunity to present this work. Okay, so I'll be presenting this report of the ferromagnetism in thin fullerene films as a consequence of 600 kV carbon ion irradiations. Okay. Outline of my talk will be ion-matter interaction regarding the accelerator, the dendritron microbeam, brief history of magnetism, and I'll try to summarize the work. Okay, so it's a bit of lengthy work, but I'll try to summarize. So this is the accelerator which I have used from IIT Kanpur for using ions to induce magnetism or magnetization in the samples which are normally diamagnetic. Okay. So, and this is the microbeam part where we can focus ion beam down to micron size. Okay. And this is the schematic of this setup. And this one is the actual setup. And here samples are loaded. And beam is focused down to 500 nanometers. And ions you can choose from atomic number 1 to 6. Okay, like proton to carbon. You can choose higher, but bending them into this microbeam line is a problematic. So I'll briefly discuss about the brief history of magnetization in carbon-related materials. So there are many more literature, but I'll stick to only few. So earliest report of weak magnetization in carbon-related material was in this TDA, okay, this like charge transfer salt, okay, of this tetrakis dimethylaminoethylene, okay, and it had a Curie temperature of around 16 Kelvin, okay, and that was in 1991, and then it got the attention of people, and this shows you the the M versus T curve, okay, clearly you have this peak, okay, and you also have this, so that tells you about the Curie temperature, and this one is M versus H. In these samples, the coercivity, that was the basically the, if you zoom it up, there is a very small coercivity, okay, but still the signatures of magnetization in samples were visible, that too below 16 Kelvin. So nowadays, there are a lot of literature available that, as I told you, that why is it exotic, and it is also known as a D0 magnetization. Okay, normally like F and D transition metals are known to be ferromagnetic at room temperature, okay, or any kind of magnetization they show. Only F group and D group element, and these are the elements which are basically like a carbon. So it is last electron will be found in P cell. Okay, so and they are not supposed to be magnetic in nature. So that is why they are known as D0, because there's no electrons available in like D cell. Okay, so they are also known as D0. That was the exotic thing, and a lot of report has been given. So there are three usual suspects of the magnetization. One is vacancy creation, one is interstitial defects, one is hydrogen chemisorption. Okay, so this vacancy creation, there is due to vacancy, there is uncompensated magnetic movements, and that leads to a Bohr magneton value, okay, of magnetization near the vacancy, and same goes for interstitial sites. And the hydrogen chemisorption, suppose some hydrogen gets bonded, okay, with the molecules, so that dangling bond, okay, that basically because of the presence of the dangling bond, that has an extra magnetic moment, and if you have more of such correlations, you will have exchange interaction. And if you carefully craft these kind of defects in the materials, there have been reports that these lead to a correlated response at room temperature ferromagnetic ordering in the samples. And first instance was shown by Eskenazi et al., a German group in Leipzig, and they showed, that was published in PRL in 2003. Okay, and so this one showed you that graphite can be magnetized using proton irradiation of 2.25 MeV, and that they showed that when you increase the dose, it goes up, and when you increase further, it comes down. Okay, so you cannot have like too much of dose, you cannot have too less of a dose. You have to have a optimized fluence. Fluence means like number of ions falling per centimeter square. So when you increase the dose, like how many ions you have irradiated per square unit of area on the sample. Okay, so that there is a threshold in these studies. So the tools which help you probing these systems are discrete magnetometer, VSM, magnetoresistance, and then there is this. So they are the magnetic signals, the first three, and rest are basically to explore about the defects in the sample, like what kind of defects are there. Okay, like when you have a graphite and when you have dangling bond, so basically you change the hybridization from sp2 to sp3, then you what kind of defects you have found, whether it is mono vacancy, di vacancy, or multiple atom vacancy. Then positron analysis and spectroscopy, that is helpful. Then you have a nuclear magnetic resonance that can also help you know about the magnetization of the samples. Okay, their nuclear Landis G number. Okay, that is, and then you have this X-ray absorption spectroscopy, XMCD, MFM, XPS, PIXI. So also, I mean, these studies, you have to tell about the impurities in the sample. Okay, so that the magnetization observed can be pinned only on the effect of the ion radiation. Otherwise, people may doubt your results. So you have to rule out the presence of this transition metal elements. Okay, even in trace amount that can be. So there is one report which we have worked, that is 1 MeV proton ion irradiation, and you can see before irradiation, this is the M versus H. It is purely diamagnetic, and after irradiation, what you see, there's a clear, there's a huge ordering that has been achieved. So here you have this fullerene structure. It's like a buckyball, and when you ion beams are impinged upon, you can have dangling bond vacancies and various kind of thing, and their correlated response can lead you to magnetization like this. Okay, this is again the extracted form, unirradiated, and after a fluence, what you see. And this is the correlated response at various doses, 5.88 picocoulomb per micrometer square. So this number of ions per centimeter square can also be converted to charge, total charge per unit area. Okay, that conversion is very straightforward. So you can see at 29.4 picocoulomb, it is maximum, and then it comes down. Okay, and this is also reflected in this study. What you see, like here, this total charge versus suggested magnetic moment, that only at certain total charge value, the magnetization is maximum. Below and above, again, it comes down. And this is vacancy density versus target depth. So you can see, like with respect to various doses, how vacancy density changes in the sample. Okay, and so with that curve, previous curve, we have calculated how much distance between defects will be there with respect to the total charge irradiated. And that curve, you can see, like in the beginning, when the total charge irradiated is less, the defects are far away, and when you keep increasing the dose, the defects come closer, and when they come too close, then also sample is amorphized and you will not have any correlation. So for this effect to develop, your sample should not be entirely damaged. Okay, that is why you have a proper window that below and above saturation magnetic moment basically diminishes. So we have also done this study with 600 keV carbon ions, and the idea was that this carbon can also prove to be useful because it can create vacancies, it can fill vacancies as well. So the effect we have done with these experiments at 2 Kelvin and at 300 Kelvin, and you can see like this carbon ions, they give you success at only the first dose. Okay, and that is the red one. Okay, and then beyond the other doses it comes down. Okay, so here even with first dose you go, magnetization observed is more, and then it comes down due to excessive damage because carbon is heavier. Okay, so here this is the XRD analysis. This is the unirradiated sample, then irradiated one. You can see only the first sample, the crystallinity is preserved. Rest samples, the peaks are gone. Okay, the 2, 3, 4, 5, you can see, and this is explained in this M versus H curve as well. So the structure of the fullerene has been broken completely. Okay, by the excess dose, and that is needs to be basically simulated as well. What we have done, and we have also found that figure, that nature in the Raman analysis. Okay, where these the orange curve tells you about the unirradiated samples data, and the black one tells you about the first dose, and beyond that all the structure of the fullerene is gone. So that is confirmed both in XRD and this thing. So I'll conclude my talk that this ion beam induced controlled damages can be utilized to induce ferromagnetism in fullerene films at room temperature. Okay, carbon ions are relatively not very effective in terms of induced magnetization sample, but we can tune the energy further to know about the outcome, and it is inducing magnetism in, I have talked about it, that non-transition group elements like sp cell electron elements. Okay, basically D0 as fullerene graphite is of immense interest in recent years, and a lot of research activity is going on, and basically they can be used as a memory devices. Okay, for data storage, and it is also biocompatible. And so one of my students will also present on MOS2. Maybe I will have to present another session. So these are some key publications. And I have also not explained because time was less. So one paper in carbon that was in HOPG, we have induced very good amount of magnetization, okay, using carbon in HOPG. But time was less, so I did not talk about that. So I thank you for your kind attention, and I welcome any questions.
Thank you for the presentation, Leader. Any questions you can ask? One question. Time for one question. If you have any question, you can ask. Right. If we don't have any question, we will move to next presentation. Yeah, third presenter, Dr. Nubul Prasad. Third presentation, OP3, Dr. Nubul Prasad from India. You can start, sir, third presentation.
Sir, Dr. Nupur Prasad is not available, so we will go to the next presentation, Dr. Thakur Prasad.
Yeah, sure. Yeah, next presentation, OP4, Dr. Thakur Prasad here. Sir, you can start. You can start, sir. You can start the presentation.
Dr. Thakur Prasad, you're requested to unmute yourself.
Yeah. Yes. You are hear me?
Yeah, we can hear you, sir. Also, for the sake of recording, we want you to enable your video.
Yeah. But I think my video is not working. I think maybe I don't know.
Okay, no problem, sir.
Yeah.
You can
Yeah.
continue.
Yeah. Okay. Thank you, Chairman. And I would also like to thanks organizer for providing me this opportunity to say something about my work. So my work is basically synthesizing of two-dimensional materials, and here two-dimensional material is uh, a kind of alloy system. So this two-dimensional alloy system was used for renewable energy applications. So if you see this graph, and also demand of energy. So demand of energy is increasing day by day, and especially in the developing country, if you see the situation of China and India, and the energy demand is quite high if you see here. But in developing country, the energy demand is already saturated. If you compare the production, so production is also going to saturate, because conventional energy source like natural oil, natural gas and coal, so it will finish very soon in future. So we need some alternative energy source. So even we have this kind of energy fuel, so it will create lot of pollutions. If you see here in this curve, so the all kind of fuel, hydrocarbons fuel, like liquid or solid or gas, you can see here the pollution is going increasing day by day, and this pollution is creating global warming. If you see this picture, by this picture the CO2 layer is there in the atmosphere, and this will absorb the infrared radiations, and this create the global warming. The effect of the global warming you can see here, the rising the sea level day by day, and the oxygen depletions, and also the water resource depletions. And if you see the climate change direct effect of the global warming, so in Thailand the whole city was flooded in 2011, and melting of the glaciers everywhere in Himalaya region also, and Alaska mountain also. So this global warming is nothing but you can say that this is the future terrorist of the humankind. If you see in India also the effect of the global warming, flooding is very common nowadays. And if you see here in this picture, the Cochin flood in 2018, it was affected much of the whole city. And you can also observe the direct pollutions by the using the hydrocarbon energy sources. This is also very dangerous, and every time you are using mask, of course in corona time also we are using mask, because so it appears to me that we have to use mask every time. So why we need some alternative energy sources to reduce the global warming effect or pollution effect, and also the independency of the hydrocarbons, because hydrocarbons will finish very soon. So there is a lot of energy sources, for example solar energy sources. Dr. PK Singh is also working in the area of solar energy, wind power energy, ocean wave energy, hydropower energy, geothermal energy. These are different energy sources is there, and last you can see the hydrogen energy. So my interest is basically related to hydrogen energy. And what is the beauty of the hydrogen energy? If you see, the hydrogen energy can be produced of burning of the hydrogen in the presence of oxygen, and we can get some exhaust energy here, for example here, and we will get water. Again we can split the water, and we can get oxygen and hydrogen, and this hydrogen will again burn in the presence of oxygen, and we will get the water. If you see the hydrocarbon content, so there is no any carbon source is there in this energy source. And if you compare the fuel efficiency, for example here, so hydrogen fuel efficiency is 33.33 kilowatt per kg, where the petrol fuel efficiency is 12.4 kilowatt per kg. So fuel efficiency is almost triple if you compare the fuel efficiency with respect to the petrol. And also the toxicity, if you see here, in hydrogen there is no toxic materials, there is no carbon content. But if you use methanol or petrol, other hydrocarbon energy sources, so you will get toxic material and also pollutant materials. Carbon, and this carbon will burn in the presence of oxygen, will make the CO2. But where is the problems? Where is the problems? Though production of hydrogen is one issue, and storage of hydrogen is different issue. So I'm working in the both area, production and the storage, but I will discuss only about the production here. So we have tried to synthesize two-dimensional metallic alloy, where we have used uh, two-dimensional metallic alloy as a electrode for hydrogen productions. So this is the different examples of the different kind of nanomaterials, which is already discussed from yesterday. So I'm not going to discuss about details. What is the beauty of two-dimensional nanomaterials? Because the Daltoning bond of the two-dimensional nanomaterials is different compared than bulk, and this will create different kind of energy scenario of the surface materials. So it will react very fast compared to the bulk. So this is the beauty of two-dimensional nanomaterials. So I'm just trying to synthesize the metallic two-dimensional nanomaterial first time. So this is the one example of the metallic alloy where we have chosen the quasi-crystalline material as a precursor materials. So if you see here in this graph, if you arrange the square, lattice, or rhombus or hexagon, it will nicely arrange. But if you arrange the pentagon, some gap is always there. For example, here, and in 2020, one of the discoverer of this five-fold symmetry mathematically got the Nobel Prize, but not for this work, for different work. Roger Penrose Stirling is here. So he has discovered theoretically the quasi-crystalline material five-fold symmetry with the help of some tiling, and this tiling is known as Penrose tiling. Actually, quasi-crystal was discovered by Setman in 1984. The paper was published in 1984. Actually, the quasi-crystal was discovered on 4th April 1982. So he has discovered the quasi-crystalline materials. It is a kind of alloy, aluminum 86 Mn14, and this alloy shows the five-fold symmetry. And further, in the same kind of alloy with different compositions, another kind of symmetry was also discovered, say the decagonal quasi-crystalline material. So we have choose both example in our present studies, the icosahedral quasi-crystal and decagonal quasi-crystals, and we have tried to synthesize two-dimensional layer materials, two-dimensional layer alloy in the both kind of systems. So here you can see we have chosen the aluminum, palladium, manganese. This is the alloy compositions of the icosahedral phase, and we have exfoliated these materials. For example, this is a kind of alloy, aluminum, palladium, manganese, in DMF solutions for 30 to 60 hours. And after that, we have got these kind of flags. If you see here, this kind of flags in the DMF solutions, and this flags was further characterized with the help of different kind of characterization techniques. For example, if you see the XRD picture here, and compare with the bulk. So bulk XRD is here, you can get all the phases, all the orientation is there. But if you see in the exfoliated sample, only particular plane is there. So only particular plane is there. We have further characterized this particular plane with the help of TEM. This is the X-space characterization techniques, and this is the SEM picture of the bulk and also two-dimensional alloy materials. If you see here the FM picture, you can see here the thickness of this exfoliated thin sheet is around 4 nanometers. So if you see the TEM characterizations here, so you can clearly see here the flags of triangular flags is there. This triangular flags is nothing but threefold orientation of the quasi-crystalline materials. So further characterizations, aperiodic nature you have already observed here by with the help of scanning tunneling electron microscope, dark field image and bright field image. If you see the modeling also, the aluminum, palladium, manganese, the aluminum, palladium, manganese, we have tried to put aluminum, palladium, aluminum, manganese, and aluminum, palladium, manganese also here. So the aluminum, palladium, manganese is the most stable materials in this case. So why this exfoliation is taking place? So if you see the cluster of icosahedrals, so it looks like the arrangement of 20 similar triangles. And when you are putting some energy in in the ultrasonic bath, the energy will transfer on the edges of this triangle and you can see this similar type of triangles flags in the exploded sample. This paper was published in Advanced Functional Materials, and this is the different calculation and try to see the stability. I am going to conclude because already chairman indicated. So another paper was published in ACS Nano, and we have tried to see the decagonal quasi-crystalline materials, and in same way here we have chosen aluminum, copper, cobalt, and we have experimented in the same type of experiment DMF solutions, and we have got the 5-fold symmetry and also 10-fold symmetry. And this 5-fold symmetry and 10-fold symmetry is also in a very thin layer. For example, if you see here the AFM picture height distributions 2.5 nanometers. So in this process we have synthesized the aluminum, copper, cobalt two-dimensional Li, for example 2.5 nanometer thick Li. So it was further characterized with the help of TEM, and we have tried to mix this material, two-dimensional materials, with two-dimensional WS2, and we have tried to make heterostructure, and we have further analyzed with the help of TEM and also XPS. And further, we have tried to see the hydrogen evaluation reactions. So we have got one 60 times higher hydrogen evaluation efficiency compared to the bulk. So these two-dimensional Li materials can be used for hydrogen production in HER reactions, and which is almost close to the platinum electrode. So our conclusion is we have realized the first time large-scale synthesis of two-dimensional icosahedral quasi-crystal and as well as decagonal quasi-crystalline materials, and we have tried to see the stability of these two-dimensional LIS with the help of ab initio calculation, DFT calculations, and we have tried to see that in icosahedral case, the 3-fold symmetry axis is the most stable, whereas in the case of decagonal quasi-crystals, the 2-fold symmetry and 10-fold symmetry is the most stable phase. And this material can be used for hydrogen production in a scalable way. Thank you for your kind attention. So I would like to acknowledge some pupils. These are the group, and we are very sorry to inform you that our group head, Professor Rohan Srivastava, was expired on 24th April 2021 due to Corona. And thank you for your time.
Yeah, thank you, Dr. Thakur, for doing your nice presentation. Yeah, we don't have enough time for questions. If you have any questions, you can contact him personally. You can ask him.
Thank
Yeah,
you.
next, yeah,
Thank you.
yeah, next presentation is actually OP3. Kavita, you can present. OP3.
Thank you, sir.
Yeah,
Is my screen visible?
yeah, visible.
Okay, thank you, sir. Myself, Kavita Kalra. I'm a student of PhD from LPU Fagwara from Punjab under the supervision of Dr. Nupur Prasad, and my co-supervisor is Dr. Vandana Chabla. My topic of presentation is role of phytochemicals in the synthesis of zinc oxide nanoparticles. It is a mini review out of the concerned literature I have gone through.
Miss
And
Kavita, can you
yes,
please make it full screen?
okay.
And enable your video.
Sure, ma'am. Sure, ma'am. Sorry. Now, is it visible? Is it okay?
Yes, it is visible. Yeah. Thank you.
Thank you. So my talk outline will be like this. First part will be introductory to nanotechnology. Then I will later be discuss about the properties of zinc oxide nanoparticles along with the applications of these particles. Then next I have enlisted a few of routes of synthesis of these particles. Then I'll talk about phytochemicals and ultimately the role in the synthesis of zinc oxide nanoparticles. And ultimately, I would like to summarize my topic. And of course, I have, and last slide, I have added some of our important references. So to start with, just to have the introduction of nanotechnology, we know it refers to the synthesis, development, characterization for the processing and applications of new systems, structures, and nano platforms. Nanomaterials, they are the particles ranging in the size of 10 raised to the minus 9 meters that differ from bulk material due to their nanometric scale. They are actually very small particles, modified forms of basic elements derived by changing their atomic and molecular properties. Nanomaterials, they show distinctive physicochemical properties when compared to their bulk counterparts. The novel properties of these nanoparticles, they are derived due to their change in their size, distribution, and morphology. And in nanoscales, they are manufactured by controlled manipulation of size and shape of particles. As Dr. Thakur just have told about the importance of the properties dependent upon the size and morphology of the particles. We know the properties of nanoparticles, they are dependent on their size and shape and structure. So application-based nanoparticles of desired morphology are being obtained nowadays. So based upon their morphology, nanomaterials, they have been classified into mainly 4 groups as 0 dimensions, like fullerene, for example, then 1 dimensional in the form of nanorods, nanotubes, and nanowires, for example, carbon nanotubes, then 2 dimensional, for example, graphene, and 3 dimensional structures like graphite, for example. So I would like to go a little bit in detail of the properties of the zinc oxide nanoparticles. I have chosen these particles out of all the metal nanoparticles. Different type of, metallic and non-metallic nanomaterials like copper, silver, titanium, magnesium, silver, iron, gold, and copper. They are being used in nanotechnology these days. Metal nanoparticles, they have gained considerable interest in various areas of science and technology. And out of all the metal oxide nanoparticles, zinc oxide nanoparticles have been considered to be the safest, easy to be synthesized, and inexpensive in their preparation. They are low toxic, they are eco-friendly, and they have been enlisted as GRAS, GRAS, generally recognized as safe by the Food and Drug Administration, US, United States. And they are being extensively used in the discipline of physics, chemistry, biology, biochemistry, and engineering. And they've got attention to their unique antimicrobial, then biocompatibility, then high catalytic UV filtering properties, high refractive index, antioxidant properties. I would like to discuss only briefly. For example, UV absorbance property of zinc oxide nanoparticles is rare, as when they are exposed to UV light, they show high absorbance in their spectrum. Similarly, they show high refractive index. The catalytic property increases due to increased surface area to volume ratio at the nanoscale, and due to their high aspect ratio, these nanoparticles will show enhanced reactivity, or we can say the catalytic activity, which is very important in many reactions or synthesis of many other nanomaterials. Then regarding biocompatibility, very good, very important property have to my interest. They have proven to be biocompatible to human body because they are safer, they are less toxic, rather they are required by the human body as nutrients in elemental or the molecular form, and it is an essential micronutrient for humans as well as animals as well as for plants. So zinc oxide nanoparticles, they show biocompatibility as well up to certain levels of concentration, we can say, to the human bodies or animal bodies, or even to the plants. Then they are very good antioxidants. And antioxidant, we know, is a chemical compound that protects the living cells against the effects of the free radicals. Free radicals, these are the, we can say, unstable molecules made up by the process of oxidation during the normal metabolism. And briefly, very important property I find, that is their antibacterial property. And zinc oxide nanoparticles, they have unique physicochemical characteristics that can affect biological and toxicological responses in the microorganisms. So they kill bacteria or microbes either by their direct attack on them, by puncturing the layer of the sugars, proteins, and lipids present in their cell wall or cell membranes. And this causes the breakage of the cell wall and cell membrane, leaking the cell contents, causing their death. Or by releasing the zinc divalent ions, which enter the bacteria themselves and hinder their growth by hindering the metabolic processes, causing their death. Or third mechanism can be that zinc oxide may produce ROS, means reactive oxygen species, for example, like hydrogen peroxide, which combine with the protein or lipid bilayers of the bacteria and destroy the cell membrane, leaking again the cell contents, leading to their death. And based upon these properties, they have many applications. A few of them I have chosen for, like based upon their antibacterial property I just discussed now, they can be used for water purification. As they release oxygen species from their surface, which causes the fatal damage to the microorganisms by damaging their DNA or cell membranes or cell proteins. Various other treatments like chlorination, ozonation, and UV treatments, they have their other limitations. For example, chlorination process is unable to kill the very resistant bacteria and all that. And depending on the same property, that is antibiotic or antimicrobial property, these have many applications in biomedical fields. They are used in the treatment of various kinds of cancers like colon cancer, lung cancer, ovarian cancer, because they generate again the reactive oxygen species and induce the mitochondrial mediated apoptosis, or they may arrest the cell cycle, maybe in the growth phase or during the metaphasic phase during the cell division. So they kill the cells, the cancerous cells, and they are selective in their nature. They don't harm the other normal cells. That is very important property I find. And they can be used as antibiotics for sanitization and sterilization. That is in the coatings of the surgical instruments. Based upon the same antimicrobial property, they can be used in ointments, lotions, mouthwashes, in dental implants, and so forth. Then they effectively absorb the UV light, but they are transparent to the visible light due to a large band gap. So they protect the skin from the harmful effects of the UV radiations, and those are used in cosmetics. So they are less adhesive, maybe easily rubbed on the skin. That's why they are very common. They're very famous to you for their use in the cosmetics. And one more important or interesting property I find, that is their role in the drug delivery and in plasma DNA delivery. They are capable of enhancing many important
Please
properties of.
conclude. Please conclude.
Yes, sir. Sorry.
Please conclude.
Okay, thank you, sir. So they are capable of enhancing many important properties of free drugs, as in maintaining the in vitro stability or solubility and their biodistribution. So they can be used in the drug delivery systems. 3D or three-dimensional tetrapode-like zinc oxide nanostructures have been synthesized, which can act as carriers for the mammalian cell transfections. So they may behave like phages, means like bacteriophages, delivering the plasma DNA or a few genes into the cells. Then due to their properties such as high permittivity and low temperature of processing, they can be used in electronics. So briefing this slide, they play a very important role in agriculture, being an essential element of plants. Zinc is absorbed as a divalent through the roots, and there's an increasing interest of its use in the agronomy. They show increased effect on growth and yield of the plants in biomass production. They're resistant to the drought stress in the plants, increasing the pollen viability, increasing the transportation rate in roots and shoots. And along with that, they can be used as fertilizers, herbicides, pesticides, insecticides, as well as fungicides to improve the yield of the crops. Then I have enlisted a few routes of the synthesis because they are being exploited very much in so many commercial fields. So their synthesis is very important. I have only categorized or classified these or enlisted these routes of synthesis into mainly divided into physical, chemical, and biological methods. Very common methods in physical, they come ball milling, laser ablation, thermal decomposition, sputtering, and solvent decomposition. And very common I have listed over there. Sol gel in the chemical method, and co-precipitation is very common, pyrolysis, wet etching, and solvothermal, etc. There are much more, but I haven't listed only a few. And interesting to my interest, very common, or I can say safer method I find is biological method. They can be zinc oxide nanoparticles can be synthesized from the living organisms like from bacteria, fungi, algae, plants, and wastes. Then oh, sorry.
Please conclude. No time is
Okay.
more than.
Excuse me, sir. Now, ultimately, phytochemicals in the synthesis of the zinc oxide nanoparticles from the plants or plant extracts, they play a very important roles. So they are the wide variety of non nutritive chemicals compounds we can say found in the plants. foods which may have health effects. They are found in vegetable fruits, nuts, and may help slowing the age and reduce the risk of many diseases, including cancer disease, stroke, high BP, cataracts, urinary tract infection, etc., etc. And the manufacture of metal nanoparticles involves three
Please
main components.
conclude. It's more than 12 minutes. Please conclude.
Okay, okay, fine. So I have enlisted the phytochemicals into main 5 types: alkalides, phytosterols, polyphenols, terpenoids, organosulfurs. These are the examples I have tabulated. Try to tabulate the examples of these phytochemicals, and ultimately, concludingly, the very important roles of the phytochemicals which they play in the synthesis of zinc oxide nanoparticles as surfactants, which reduce the surface tension of the liquids in they are dissolved, as capping agents, coating the nanoparticles, as reducing agents to oxidize themselves, and they reduce to the other reactants, as stabilizing agents, stabilizing the reaction products formed, that is the nanoparticles formed, and as well as chelating agents, which form the multiple bonds and facilitate the process synthesis of these nanoparticles. So to summarize briefly, nanotechnology is an umbrella term, and I try to incorporate the properties as well as the applications of the zinc oxide nanoparticles, and uh, I concluded with the phytochemicals roles during the synthesis of the zinc oxide nanoparticles. So ultimately, in the last slide, I have tried to add very important references. Thank you very much for giving your attention. Thank you.
Thanks for your presentation. Actually, next presentation OP5 is not there, John Kumar Reddy. Now I am going to invite sixth presentation OP6, Dr. N. Raju. Dr. N. Raju.
Yes, sir. Yes, sir. I will.
Dr. N. Raju, please share
Yes,
your
sir.
screen.
Yes, sir. Sir. Sir, how to share, sir?
Yeah, actually at the bottom they have, we just can observe green color, share screen.
Where, sir?
At bottom.
Bottom.
Yeah, share after chart, between chart and record, share screen is the option.
It's not. It's not coming, sir.
Share screen option is there at the bottom. Any problem with sharing?
Dr. N. Raju, can you please see the bottom? At the bottom, there is a share screen option in green in your application. Are you doing it from your laptop?
Yes, sir. Yes, ma'am.
There at the bottom, you will see share the screen option. Can you see that?
It's not showing, ma'am.
If you see some dots, then maybe you click the dots. Dots will give you more options.
It's not showing.
Okay, so can we take your presentation after some time?
Yes, ma'am. Yes.
Yeah.
Yeah.
Okay, now I'm inviting next presentation, OP7, Shreya.
Instead of OP7, कौन सा switch हुआ?
Shreya is there? Next presentation, Shreya Matel, OP7.
OP7, instead of OP7, sir, please make an announcement for OP12.
OP12. Okay. Yeah.
OP? Sorry, OP20. OP20.
Okay.
OP20 will make the presentation now.
Okay. Okay. Sudeep Chakrabarti. Yeah, you can start your presentation. Sudeep Chakrabarti. OP20.
Am I audible, sir?
Yeah, audible.
Yeah, Sudip Chakrabarti here. Okay, sir, I'm going to share my screen. Visibility, sir?
You can, yeah, right. You can start now.
Okay, sir. Thank you for giving me this particular opportunity to present my research work in this conference. So basically here, I'm going to, talk about structural parameters of air wing based off NACA 2412 profile.
Screen share cheyadante.
And CFD study has been done here in order to get the results. And we have used different composite materials, right? So myself, Sudip Chakravarti, and along with me, Mr. Somaghas, who is working as assistant professor in the Department of Mechanical Engineering at Adamas University. He is one of the contributors of this particular research work. So the three things which are most important in order to describe any kind of aerodynamics, the first one is a lift, drag, and the weight of this particular vehicle. So we are trying to incorporate. So someone has raised the hand, Ankit sir, I believe. Should I go with my presentation, sir, or should I stop?
Yes, please continue, sir.
Okay, thank you. So now here we have tried to incorporate, like few composite materials which will be responsible for, lowering the body weight of the aerodynamic vehicle, and this will be giving us this particular ease to increase the lift and drag force associated to it. And we know if you can increase the lift force, by putting the same amount of fuel into the aerodynamic vehicle, then it would be even better for any kind of vehicle to be used, and the efficiency will be increased in this case. So the three different materials we have used over here, the first composite material which we have used is carbon fibers, and the second one is the alpha beta titanium alloy, and the third one which we have used is a aluminum zinc magnesium alloy. And the entire work has been done in ANSYS workbench. And here in India, many researchers, they do not accept the result of the ANSYS. For this reason only, we have done the grid independent study for showing the convergence of this particular research work. So this is the abstract which I have shown you on the screen in front of you, and the objective of the research work is to investigate the structural parameters, such as few structural parameters we have taken, that first one is a von Mises trace, the second one is a total deformation, and the third one is a von Mises strain. And along with this, the total heat flask and the mode shapes are to be analyzed in this particular work by using different composite materials. And why I am choosing this particular aerofoil, which is NACA 2412, because NACA 2412 is the most commercialized aerofoil in this decade. And the second objective I'm having, that to identify the acceptability of carbon fiber, alpha beta titanium alloy, and the aluminum zinc magnesium alloy in the aerodynamic structures. So moving forward with this particular presentation, this is my motivation of the work, which I have portrayed on the screen. And the thing is this, again, I am telling you that for increasing the acceptability of the research work, for increasing the acceptability of the research work, we have done the MES independent study for showing the convergence of the study, for getting the all the output parameters more accurate. So this is it. So this graph we have got from the MES independent study, and the output which we are getting from this particular research work will be acceptable in the industry as well. So these are the methods by which we have carried out this particular research work. The first, we have done the modeling in ANSYS workbench in the design modular field, and after that we have done the meshing. After that, formulation of properties to each element and concern nodes, give inputs to the system such as load, force, temperatures, and after that we have solved by putting the boundary conditions. So these are the properties of the structural properties of the structural, as well as few thermal properties I have included according to my work. So and I have tabulated all these things here, in front of you, and the three different composite materials which I have taken. I have segregated the three composite materials, based on the density, Young's modulus, thermal conductivity, and the Poisson ratio. So this is it. And after that, these are the simulation result. So first we are going to deal with the total deformation. So we are going to find out the total deformation for three different materials which we have used for this particular air wing. So these are the range which I am getting. So for one instances, I can easily conclude that for carbon fiber, the first one, I am getting the maximum deformation of 0.001961 meter. Pardon me. And for the second one, the alpha beta titanium alloy, I am getting the maximum deformation as 0.0003803 millimeter. So as per the deformation, because here the load which I have applied to all the three composite materials will be uniform, will be same. By keeping the load constant, this deformation I have get for I have got for the three different composite materials over here. And after the total deformation part, we have simulated the total heat flux amount. So the total heat flux amount has been simulated for three different composite materials which we have taken for our research work. So this is the result. So this is a simulation result of it. And after this, that von Mises trace has been analyzed. So in terms of Newton per meter square Pascal, so all the stresses we have analyzed over here for the different three composite materials. And after the von Mises trace, we have got, and we have simulated the von Mises strain as well. So this is the graph of it. So, and after that, the mode shape, the most crucial one, because until or unless we have got the mode shape, we cannot conclude that at which frequency this particular air wing can be operated. Suppose if the outer frequency is matched with the inner or the structural frequency of this particular air wing, so we know that a resonance can be formed, and which is we can say the undesired thing for any kind of structure to be operated. So here first, for the first figure, we have imparted 17.685 hertz, and if I import this particular frequency, the mode shape we can see like this. And if I increase the frequency level from 17.685 to 80.935, we can see that particular shape has been changed, and this will be giving you the alarming condition that for this particular frequency level, what can be the deformation level. So this is the relation of it. So this is the most important thing we need to understand, that this is the mode shape for the alpha beta titanium alloy. Here we have taken three different range of the frequency. The first one frequency which I have taken as 808.04 hertz, and the second one frequency which I have taken as 3659.1 hertz, and the third frequency as we have taken, as you can see on the screen, it is 3659.1 hertz. Now here you can see, for the first figure, you can easily conclude that if I impart 808 hertz to this particular composite material which is alpha beta titanium alloy, then we can see that this particular wing will not be operated so easily. The structural that breakdown we can easily experience by imparting such a frequency to this particular structure. So this is the last one, is that mode shape of the aluminum, zinc, and magnesium alloy. So the same work has been done on this particular composite material, but here the lower frequency range we have chosen. The first one we have imparted here as a frequency level of 80.935 hertz, and the second frequency level which I have put over here as 17.685 hertz, and the third I have put as 103.23 hertz. And here I can easily conclude that if I impart 103.23 hertz, that mode shape will be different, and this cannot be operated so easily. So now coming back to the next slide. So here I have tabulated that properties in terms of the total deformation, total heat flux, von Mises trace, and the von Mises strain of carbon fiber and aluminium, carbon fiber and alpha beta titanium alloy. As because see from this particular calculation, I have omitted that particular acceptability of aluminium, zinc, magnesium alloy, because this is not giving me a better result compared to the other two. So the rest of the analysis has been done by taking the other two composite materials, and the particular value which I have got out of the simulation I have tabulated over here. Okay, and this is the conclusion in front of us, that like after comparing every data which I am getting from the simulation, we can conclude that by doing the finite element analysis and the simulation, that alpha beta titanium alloy is the best fitted material as a material for the aircraft wing. So this is all from my end. This is a reference, as you can see.
Thank you for your presentation. Okay. If you have any questions, you can ask. We have time for one question. If any questions, you can ask. We have time for one question. If you have no questions, we can move for another presentation. Next presentation, OP8, Dr. J. Anthony Rajan. Next presentation, OP8, Dr. J. Anthony Rajan.
Yes, sir.
Yeah, you can share your screen.
But mine is over.
Is it visible, sir?
Is it visible?
Dr. Reddy, sir, mine is over.
Yeah, yours over, sir.
Okay, sir. Thank you very much, sir.
Okay.
Excuse me, sir.
Yeah, the screen is very large. Your
Yes, my screen is visible, sir.
But it's visible, but it's very large.
Shall I keep this, sir? Shall I keep this like this?
Yeah, you can keep like that.
And okay. Okay. Good afternoon to all. Thank you for giving this opportunity to present my paper. My topic is fabrication of electrocatalyst for oxygen reduction using silver nanoparticle modified GCE in 1,4-naphthoquinone derivative. To introduce the paper, the reduction of oxygen is an important reaction in fuel cells and biosensors, but it is not easy. So the reduction of oxygen is not easy. So we have to apply some techniques for this electrocatalyst used here. So 2-hydroxy-1,4-naphthoquinone is the most important compound for electroanalytical investigations due to their strong absorption characteristics on various electrode materials. So that we have chosen this 2-hydroxy-1,4-naphthoquinone. Then modified electrodes played a significant role in the electrocatalytic reduction of oxygen. And then nanoparticles and nanotechnology have increased its attention in many applications due to their interesting properties. So we have chosen silver nanoparticle as an electrocatalyst. Next, experimental methods. 2-hydroxy-naphthoquinone were purchased from Sigma-Aldrich. HPLC grade ethanol and double distilled water was used throughout the experiments. And then all the buffer solutions are prepared from the chemicals purchased from Merck Chemicals. Then 99.99% pure oxygen and nitrogen were used for the experiment. Working electrode, we are using 3 electrode system containing silver as a working electrode, platinum, silver as a reference electrode, platinum as counter electrode, and glassy carbon electrode with the surface area 0.0314 centimeter square were used as a working electrode. Then results, we have studied about the electrochemical behavior of 2-hydroxy-naphthoquinone by using the nature of process, effect of scan rate, and studying the effect of pH. Then electrocatalytic reduction of oxygen were studied by carrying out effect of pH and effect of scan rate and calculating the surface coverage value of naphthoquinone. Then the techniques used were cyclic voltammetry, chronoamperometry, and chronocoulometry. From chronoamperometry, we have calculated the diffusion coefficients of naphthoquinone, and from chronocoulometry, we have calculated the number of electrons involved in the reduction of naphthoquinone as well as oxygen. So this is the cyclic voltammogram of 2-hydroxynaphthoquinone at silver nanoparticle modified glassy carbon electrode in pH 7. So when So cyclic voltammogram of 2-hydroxynaphthoquinone under deaerated condition, it gives a single redox couple. And then when increasing the scan rate from 20 to 20, 50, 100 up to 700 volt per second, it gives, there is a large separation between this cathodic and anodic peak. It indicates the process is the quasi-reversible reaction. And the nature of the process is determined by the slope value of log IPC versus log nu. If the slope value of log IPC versus log nu is greater than 0.5 means the process is absorption controlled process. And also IPC versus nu is also the straight line. And also the slope value of log IPC versus log nu, which is less than 0.5 means the process is diffusion controlled process. And also IPC versus root nu gives straight line. In our study for 2-hydroxynaphthoquinone, the effect of scan rate log IPC versus nu gives straight line, and also log IPC versus log nu gives also straight line with the slope value 0.321, which is less than 0.5. The process is the diffusion controlled process. Next, the effect of pH. We have carried out the experiments from the pH is 1 to 13. So the effect of pH, from the effect of pH, but pH potential diagram, we can find out the number of electrons. If the slope value is 90 millivolt, around 90 millivolt means the process is 2 electron, 3 proton process. And 60 millivolt means it is 3 and 2 electron, 2 proton process. If it is 30 millivolt means the process is 2 electron, 1 proton process. In our study, we will get up to 3. The slope value is minus 91.71. So it is around 90. So the process is at low pH, 2 electron, 3 proton process. At intermediate pH from 3 to 10, it gives 61.2, the slope value, so that the process is 2 electron, 2 proton process. And at higher pH from 10 to 13, 30.56, which is around 30. So the process is 2 electron, 1 proton process. From this way, we can selecting the optimum pH, and then we can confirm the number of electrons involved in this reaction. Next, the catalytic reduction. Catalytic reduction of oxygen is carried out by purging oxygen through the solution for 10 minutes. This is the cyclic voltammograms of 2-hydroxynaphthoquinone at silver nanoparticle modified GCE in pH 7 in the presence and absence of oxygen. Here, actually, under deaerated condition, it gives a single redox couple. But under aeration, when oxygen is purged through the solution, anodic peak is completely vanished and the cathodic peak enhances for its maximum current. It indicates the reduction of oxygen taking place. By choosing this reduction cathodic peak current, we can confirm the optimum pH. So this is the optimum pH 7 because other pH current, cathodic peak current and also shift in oxygen reduction potential when comparing other pH. pH 7 is higher oxygen reduction potential shift and also high current, cathodic current. And this is the delta E is shift in oxygen reduction potential. So the plot of delta E versus pH gives at pH 7, it has high delta E. And also cathodic peak current is also high in the pH of 7. So this is shift in oxygen reduction potential. The shift in oxygen reduction potential, that is delta E, is the difference between the oxygen reduction potentials in the presence and absence of naphthoquinone, that is catalyst. Here, the reduction of 2-hydroxynaphthoquinone at silver nanoparticle modified glassy in pH 7 occurs at minus 658.6 millivolts. But at bare GCE, actually, it is minus 1147.5 millivolts. So the difference between the bare GCE and with naphthoquinone, the difference is 400. Excuse me, sir.
Please conclude.
Yes, sir.
Yeah, please conclude. Time is up.
Sir, can you hear me?
Yeah,
here-
please conclude.
Yeah, please conclude.
Am I audible, sir?
Yeah, audible.
Yes, yes. Okay, thank you, thank you. Thank you, ma'am. Yeah. So from this, the delta E, that is shift in oxygen reduction potential, is 488.9 millivolts. So oxygen, the optimum pH for this study is pH 7. Next, the effect of from the effect of scan rate, the nature of the process, that is the reduction of oxygen is diffusion controlled process, because the log IPC versus root nu gives the straight line, and also IPC versus root nu gives the straight line with the diffusion, with the slope value 0.274. This is less than 0.5. So the process is the diffusion controlled process. And next, surface coverage value is calculated by using gamma of naphthoquinone, that is equal to Q by NFA. The gamma is the surface coverage, Q is the charge consumed, N is the number of electrons involved, F is the Faraday, and A is the area of the glassy carbon electrode. From this formula, we can calculate the value of surface coverage, that is 0.67 into 10 to the power minus 8 mole per centimeter square.
Please conclude.
And next,
Time is 12 minutes.
yes, sir. Last one, the from the chronoamperometry and chronocoulometry, we can calculate the value of diffusion coefficient of naphthoquinone. So by using Cottrell equation, so this is the equation, the slope value. From the slope value, we can calculate the diffusion coefficient value of naphthoquinone as well as oxygen. The values are given 5.38 into 10 to the power minus 5 centimeter square per second for naphthoquinone. And then this is the chronocoulometry. So from the chronocoulometry, we can calculate the accurate value of number of electrons involved in the reduction process. For reduction of naphthoquinone, it is 2.03, and reduction of oxygen, it is 1.96. So this is around 2. So the process is 2 electron, 2 proton process. So from the chronocoulometry, under deaerated condition, there is a reversible peak is obtained, but under aeration condition, the reversible process that is the reversible process is not taken. By concluding my work, the oxygen reduction process is 2 electron, 2 proton process, and the optimum pH for this study is pH 7. At pH 7, the overpotential, that is shift in oxygen reduction potential, is 488.9 millivolts. So the modified electrode reduces at lower potential than bare GCE, so that we can use silver nanoparticle modified with the GCE with 2-hydroxynaphthoquinone can be used for oxygen sensor. Thank you for attention.
Thank you for your presentation.
Thank you, sir.
If you have any questions, you can ask later. Next presentation, OP9.
Sir, OP6 is ready with the presentation.
Yes.
Dr. N. Raju, OP6 is.
Yes. I would like to share to screen.
Rajam.
Good afternoon, everybody. I am Dr. Raju from Usmania University. I would like to thank you giving this opportunity, organizing this conference.
Please maximize your screen. Slideshow. At the top, slideshow is the slideshow.
Yes.
By using slideshow, you can maximize the screen. And here, after animations, transitions, animations, slideshow.
Where, sir?
Animation.
After animations, slideshow is there.
After animation, slideshow.
At the top you can check.
On the top ribbon.
Where?
On the top ribbon. No, sir. Sir, please, yeah. Go, yeah, file, home, insert, next. The go to slide show.
Sir, design. Where?
Yeah, next.
Yeah, come right sir.
Yeah.
Yeah.
Next.
It is not.
Slideshow is there.
It is there, yes.
Okay.
Please click this. Click it, sir. First one.
First one, sir,
From beginning.
beginning.
From beginning. Okay.
Yes.
Thank you.
Sir, please keep it on time. Dr.
Okay.
Raju. Yeah.
Okay. My topic is synthesis characterization, anti-cancer and antimicrobial activities of biometallic silver and gold nanoparticles synthesized using purple non-sulfur bacterium Rhodopseudomonas faecalis. Next. Purple non-sulfur bacteria are the most diverse and versatile of all photosynthetic bacteria with vast potential for application. These bacteria preferably grow by photoheterotropic metabolism with organic substances as electron donors during their photosynthetic activity. Most species are also capable of growing autotropically with molecular hydrogen as donor. Purple non-sulfur bacteria gram-negative organism appear red, orange, and purple due to pigments used in the photosynthesis found variety of aquatic habitats, most soils, bogs, and paddy fields. It's a NC-based admission also I was done to the Pseudomonas faecalis strain in Raju MPPR in 2014. To the Pseudomonas faecalis strain NR MPPR in 2014. This is also Rhodopseudomonas faecalis strain SMR001 in Raju 2014. These are three strains was NC-based admission I was done. These bacteria are highly useful industries. Biotechnology produce biofuels and from the hydrogen which can be used in fuels and from the hydrogen himself fuel. It can be used electricity generation and also for the transportation purpose. They can be used a source of single cell protein. These bacteria are capable of producing composite nanoparticles also. They are no produce photohormones which are effective nanoparticles, effective and indolestic acid. The carotenoids are produced to show antioxidants activity also. Research objectives: isolate different strains of photosynthetic non-sulfur bacterium from various petroleum contaminated soil samples as there are no reports of the isolation from these soils to enrich. Identify and characterize the isolated photosynthetic non-sulfur bacterium by 16S RNA sequencing NC-based deposition for individual strains to screen photosynthetic non-sulfur bacterium strain for growing biomass potentials and select efficient strains for further studies to optimize certain parameters culture conditions for the growth of the selected strains. Methods for isolation phototropic battery: collection of petrol and diesel contaminated soil samples from Delhi, Malapur, Nacharam. Isolation of photosynthetic bacteria by enrichment technique by Bible finning and medium, specific medium. Culture was maintained in Bible performing finning medium and incubated anaerobically under 2000 lakhs of light. Few strains were obtained striking the liquid cultures and solid medium and colonies were picked to inoculate in the liquid medium for the further maintenance for few strains. Culture growth was observed within 10 to 12 days. This is the specific medium. I was growing. Identification and characterization isolation bacteria isolation were identified culture characterized like color, size, and shape of carbon, nitrogen source. Physicochemical, spectral, biochemical characters were analyzed. Identify isolated strains were provided by Verghese manual system bacteriology. NCBS deposition their axial numbers were in strains. This is the I was picked soil samples. These filling stations, area of search sampling locations, Delhi, Karolbagh, and India, Hyderabad, Mallapur, and Nacharam locations. These morphology studies and characterizations of the three strains, SMR, Rhodosoma palistris SMR 001 and palistris NR MPPR, NRD. These strains are cultured. Structural characterization using TEM morphology, three strains, and UV spectrophotometry using this wavelength and absorbance. 16 years rRNA sequencing was done. This three strains methodology. DNA isolation culture using alkaline lysis method PCR was done by specific primers which amplify the bacterial strains. And agarose gel electrophoresis 2% gel. Amplification was extracted from gel using mid microgel. Extraction kit cat method. Extract produced were sequenced. This is the 16S RNA sequencing results. And synthesis composite nanoparticles. Composite nanoparticles are advanced materials having recently gained increasing attention to their scientific and technology improvement. They are find wide variety of applications such as catalyzed with huge activity and specificity metal, semiconductor junctions, optical sensors, and modified polymer films for packaging. Application study as nanoparticle synthesis or synthesis composite nanoparticles. They selected bacteria isolated was inoculate 250 ml air flask containing 100 ml sharing nutrient both. The cultured flask were incubated in the rotating shaker. Silver nanoparticle as a AgNO3 solution with a constant concentration of 0.1 ml Arium chloride with concentration of the 0.1 mm. The percent pressure and using the prepared culture filter in the provide section as a reducing agent, aqueous medium, construction of AgNO3. Characterization of synthesis composite nanoparticle, UV visibility spectrophotometry, FTIR studies, and XRD, same antibacterial activity composite nanoparticles, and characterization nanoparticles, UV visible FTIR and XRD, same antibacterial activity was done. Anti-cancer activity composite nanoparticles outcome MTT assay revealed that composite nanoparticles were found inducing more cytotoxic towards cancer cell line HT29 and MCF7. These results revealed morphological changes and IC50 values composite nanoparticle against HT29 and MCF7 cancer cell lines are represented in the tables figures. Anti-cancer activity cell lines and cell culture. HEP-G2 human cell liver cancer cell line and HeLa survival cancer cell line were obtained from National Center for Cell Science.
Please conclude, sir. Please conclude.
Cytotoxic assay was done. Cytotoxic property composite against HT29 cell line and morphological changes and composite HT29C and MCF7 cell line also. Morphological changes and control you can. There is interesting for a alternative antibacterial treatment because of the improvement antioxidant protein protection among microorganisms like display AgNO3 chloride supernet and pellet culture sample showed results color formation brown, the color intensity improved with period of incubation due to the reduction Ag. Control of without silver nano conclude. I conclude the work without isolation with the isolation bacterial strains from oil contaminated soil using enriched media. Characterization studies and done spectral data 16s RRS. The isolated strains were deposited NCBI. Further studies were optimization growth in parameters and isolated strains. Optimizing hydrogen production is the isolated strains. The silver and gold nanoparticles exhibited significant antimicrobial activity against all the tested pathogenic microorganisms when compared with standard drugs, ampicillin. Hence, the synthesized nanoparticles can be used potential antimicrobial agent in near future. This is the first reporting for photosynthetic bacterium isolated from oil contaminated soil publication. Thank you. Thank you so much.
Yeah, thank you, Raju.
Thank you, sir.
Any questions you can ask now? If you have any questions, you can ask. Okay, if there is no questions, we can move to next presentation, OP9.
Thank you, sir. Thank you.
Yeah, thank you. OP9, 9.
हाँ sir.
You can start.
It is visible, sir?
It is visible. You can screen. Yeah.
Okay, sir. Good afternoon, everyone. I am Naina from Davangere University, Research Scholar, Department of Physics, Davangere, India. Working under the guidance of Dr. M.N. Kalsat sir, Chairman, Department of Physics, Davangere University. The work is done under the collaboration with G.H. Nagveni, R.M. Hudlur sir, Siddhappa sir, and Pradeep sir. Today I am here to present the colloidal synthesis of stable zinc sulfide quantum dots. Before entering to real research work, let me give some introduction about quantum dots. Quantum dots are nothing but tiny semiconductor material whose size range lies between 2 to 10 nanometers. Nanostructures frequently show behavior which is intermediate between the tough macroscopic solid and an atomic system. Among these colloidal nanoparticles known as quantum dots. Quantum confinement effect. Quantum confinement effect is observed when the size of the particle is too small to be comparable to the wavelength of electron. If the size of the particle increases, band gap decreases. As the size of the particle decreases, we can see that there will be change in the color of the particle. As the main significance of this quantum dots is fluorescence property. Fluorescence of quantum dots. When the light absorbed by the quantum dots, it excited to higher energy state and de-excited to lower state by emitting fluorescence light. This figure shows that when the particle size increases, it looks red in color, and wavelength increases by decreasing the energy, and when the particle size decreases, it looks blue in color with wavelength shorter in wavelength and higher in energy and frequency. Quantum dots glows when illuminated under UV light. This video shows how quantum dots work under UV light. Quantum dots glows when it is under the UV light. Application of quantum dots. Quantum dots is a stage for many wide range of applications such as sensor, bioimaging, photocatalytic, biosensor, etc., nano devices, etc. Next, importance of ZNS quantum dots. The main aim of this paper is to present the colloidal synthesis of ZNS quantum dots. Here I choosing zinc sulfide quantum dots because it having larger band gap, chemically much stable, suitable for wide range of applications. Experimental plan. It will be explained using this figure. Here I took 0.5 molar of zinc acetate, and it is dissolved in 60 ml of distilled water with constant stirring using magnetic stirrer. Subsequently, 1 ml of TGA added dapwise into it, we get white turbid solution. White turbid solution. Next, add 0.0 molar of sulfur dissolved in 40 ml of distilled hydrogen hydrate. Then solution turns to white solution. After few minutes, white precipitate was settled down. Then this precipitate was poured into, washed with the distilled water until pH become neutral. Then this precipitate was poured into petri dish. Finally, powder was collected by drying. Possible reaction mechanism for the formation of ZNS quantum dots is as follows. Here, highly reactive S2- that is sulfur ion react with the Zn2 plus to form a ZNS quantum dots. This figure shows how TGA molecules, that is capping molecules attached to ZNS molecules, how affinity towards the quantum dots it shows. Result and discussion. ZNS quantum dots were successfully synthesized by chemical precipitation method. The size and shape of the quantum dot was investigated by using technique UV visible photoluminescence, XRD, and FTIR. First, UV visible analysis of ZNS quantum dots. It is a plot of absorbance versus wavelength. This figure shows optical absorption of ZNS quantum dots. The maximum absorption takes place at 313 nanometer. Using this UV visible data, we can also calculate the particle size of the quantum dots using a simple equation. Delta Eg is equal to h square by a square 8a square 1 into 1 by me plus 1 by mh. Here, h is the Planck's constant, a is the diameter of the particle, and me and mh is the effective mass of electron and hole. Here delta Eg is the energy difference between the energy gap of nanoparticle and energy gap of bulk. Energy gap of bulk ZNS is 3.72 electron volt, which was calculated by thorough literature survey, and Me and MH value, that is Me is equal to 0.34 and MH is equal to 0.23, which was also mentioned here by literature survey, and calculate the nanoparticle energy gap by using Max Planck's equation, that is E is equal to 1234 divided by lambda. Here lambda is nothing but maximum absorption, that is 313 nanometer. Substitute all this value in equation 1, we get particle size of ZNS nano quantum dots, that is 3.54 nanometers. Next, photoluminescence analysis of ZNS quantum dots. It is a plot of intensity versus wavelength in nanometer. This figure shows the emission peaks at found at 606 nanometer for the excitation wavelength of 305 nanometer. Hence, the emission was in visible spectral range. Next, FTIR analysis of ZNS quantum dots. It is a plot. It is used to study the attachment of functional group to the sample. It is a plot of transmittance in percentage and wavelength in per centimeter. Here we can see the broad banding, that is at 3468 per centimeter. This is due to OH vibration. This is due to vibration of OH, and the band 1605. This is due to bending vibration of C double bond C molecule. Next, the clear absence of SH molecule, that is thiol group, at 2550 per centimeter indicates the binding of TGA molecules to the ZNS quantum dots. The weak The weak bond situated at 656 per centimeter are allowed to the ZNS vibration. Next, XRD analysis
Please
for
conclude,
ZN.
Anna. Please conclude.
XRD analysis of ZNS quantum dots. Here obtained diffraction patterns are 28.8 and 47.63 and 56.69 corresponds to crystalline planes. Using D-Becherer equation, we can calculate the particle size. The estimated particle size is 4.29 nanometer. The observed XRD pattern is cubic crystalline structure. Conclusion. In this research work, ZNS quantum dots were successfully synthesized by chemical precipitation method and characterized by using technique UV visible, PL, FTIR, and XRD. The calculated bandgap for ZNS quantum dots is 3.94 electron volt, and estimated particle size theoretically is 3.54. And using FTIR, we can study the functional group attachment to the sample, and the cubic shaped ZNS quantum dots obtained. The prepared sample used in several applications. There are photocatalytic, sensor, LED, solar cell, etc. Thank you, sir.
Yeah, thank you for your presentation. If any questions, you can ask. We have time for one question. If any questions, you can ask. If there is no questions, we can move to next presentation. Next presentation, OP10, Nijisha. OP10.
Yes, sir. Yes.
Yeah, you can start.
So good afternoon, everyone. My work is based on the study on polyvinylidene fluoride and polyvinylidene fluoride co-hexafluoroprofiling gel electrolyte for photovoltaic application. So in this work, we are going to synthesize two polymer-based gel electrolyte, that is PVDF based and PVDF HFP based gel electrolyte. And the application, photovoltaic application, we are applying it in dye-sensitized solar cell. So quasi-solid state electrolyte or gel electrolyte, it's nothing but a polymer matrix swollen with liquid electrolyte. Here in gel electrolyte, the liquid electrolyte is expected to be trapped within the three-dimensional network of the polymer matrix. So the advantages of using this gel electrolyte is easy to synthesize this gel electrolyte. It's safe and robust. The synthesis procedures are safe and robust, and it also provides high flexibility and thermomechanical stability. So the area where these gel electrolytes are applied include DSSC, lithium-ion batteries, supercapacitors, fuel cells, etc. So we know that the first DSSC was introduced by Michael Greitzel and Oregon in 1991, and they used the liquid electrolyte, acetonitrile-based liquid electrolyte, and the efficiency they got was 7.1. And below the schematic of the DSSC operation was given. I think almost everyone might be knowing the working principle. So I'm not going into the detail of this working principle. So from the beginning itself, we know that photovoltaics, that is DSSC, depends mainly on this liquid electrolyte. We studied liquid electrolyte based DSSC. So when using this liquid electrolyte, the best performance was always given because of the high ionic conductivity, good interfacial contact, and but when using this liquid electrolyte, the shelf life of the device was very poor. Because there are a lot of drawbacks or problems encountered when fabricating this device using this liquid electrolyte. Some of them are listed here. That is, leakage and volatilization of the solvent can take place. Desorption and photodegradation of the attached dye can take place. Also, corrosion of the counter electrode can also occur. So because of these drawbacks and all, the shelf life was of DSSC with liquid electrolyte was poor. So to overcome these drawbacks, a new electrolyte was introduced, that is solid state electrolyte. But when using this solid state electrolyte also, there are a lot of problems, such as poor mobility of the charge carrier, less interfacial contact between the electrode-electrolyte interface, and hence the poor efficiency. So it became a prerequisite to introduce or to find a new state of electrolyte for DSSC, and this paved the way for the introduction of quasi-solid state electrolyte or gel electrolyte. This gel electrolyte has got the properties of both liquid as well as solid state electrolyte. It has got high ionic conductivity and good interfacial contact as liquid electrolyte. It has got cohesive nature and long-term durability as solid-state electrolyte. So, so in this work, we discusses mainly on the synthesis of polymer gel electrolyte, that is PVDF, HFP based polymer gel electrolyte. We characterize the gel electrolyte. For that, FTIR is used to obtain the structural information about the structural modification taking place in a gel the corresponding polymer. What is the change occurring when the polymer is getting gel, and the diffusion coefficient, that is the mobility of the ions, can be calculated from linear voltammetry, and the conductivity can be calculated from electrochemical impedance spectroscopy studies. And the stability of the polymer gel electrolyte for a period of 30 days was also carried out. Finally, the synthesized gel electrolyte was applied to ICSE and the photovoltaic characterization was also done. So this is the general polymer gel electrolyte preparation method. For that, first we synthesize or first we prepare the liquid electrolyte. For that, we are using electrolyte salt and mixing in organic solvent. The electrolyte salt can be sodium iodide, lithium iodide, potassium iodide, that are commonly used in DSSC. And the solvent can be any of the organic solvent like EC, PC, acetonitrile, anything. So mixing at a room temperature, we obtain the liquid electrolyte, that is the electrolyte solution. To the electrolyte solution, we are adding the polymer matrix, that is any of the polymer that of interest are added, and this mixture was mixed at elevated temperature to obtain a hot melt of polymer electrolyte solution. This upon cooling for an overnight or for some time, we get a polymer gel electrolyte. So for making PVDF based gel electrolyte, 0.3 molar potassium iodide, 0.03 molar potassium iodine, dimethyl formamide and propylene carbonate was used as a solvent. And to this liquid electrolyte mixture, PVDF, required amount of PVDF was added, and a temperature of 80 degrees Celsius was given for a 30 minutes. And we obtain a highly viscous hot melt. This was kept for an overnight, and we obtain a freestanding PVDF gel electrolyte. Similarly, for obtaining PVDF HFP based gel electrolyte, 0.3 molar potassium iodide, 0.03 molar iodine, and propylene carbonate, ethylene carbonate solvent was used, and to that PVDF HFP required amount was added. A temperature of 80 degrees Celsius was given for 30 minutes, and we obtain a highly viscous hot melt. This upon keeping for an overnight, we get gel electrolyte. This is how we synthesized both the gel electrolyte. For characterizing the gel electrolyte, that is using linear voltammetry and electrochemical impedance spectroscopy, we need to fabricate a symmetric cell. The structure of the symmetric cell is given here. So for that we have to make platinum deposited FTO as the electrode material. So electrochemical deposition is normally performed to fabricate the electrode material. So in between the two platinum coated FTO, we are sandwiching the gel electrolyte, likewise to make the symmetric cell. So in between these electrodes, we sandwich the gel electrolyte, and we record the LSV as well as EIS spectra. So the formed platinum was confirmed by the XRD data, and the crystalline spectra shows well matching with the JCPDS file number 870647, and the morphology of the platinum was obtained from the SEM image, and it showed a buttrose-like structure. So from the FTR data, here that the FTR was taken for PVDF powder, PVDF polymer, and PVDF gel electrolyte. So when looking the PVDF gel electrolyte, some extra peak was found. So this confirmed the presence of liquid electrolyte within the polymer network. So the extra peak corresponds to the presence of the liquid electrolyte, that is the organic solvent present in it. And from the limiting current potential graph, that is the LSV graph, the limiting current for PVDF obtained was limiting current density obtained was 15.56. By substituting the value of limiting current in the above expression, J limiting is equal to 2NF D apparent C by D, we obtain the diffusion coefficient of I3 minus 0.37. Similarly, from the impedance spectra, the conductance can be calculated by the expression sigma is equal to L by RBA. So the first semicircle corresponds to the resistance at the counter electrolyte interface, and the second semicircle corresponds to the Barber impedance. So from that, the R1 is the first, R1 corresponds to the first semicircle value, and R2 corresponds to the second semicircle value. And by substituting the value in sigma is equal to L by RBA, where L is the thickness of the film, A is the area, and RB is the bulk resistance, we obtain the conductivity as 2.04. A time-dependent stability study of PVDF-based electrolyte was recorded for a period of 30 days by recording the LSV, and we can see that the limiting current density for the first day was found to be 15.56, and it degraded to and it degraded to 6.88 when the days passes and it reached as we reached the end of the month. Similarly, the diffusion coefficient value also degraded. So a decrease in performance with the time was due to the destruction of the structure of this gel electrolyte. So for the same set of characterization was performed for PVDF HFP based gel electrolyte also. Here also the presence of liquid electrolyte in the network of polymer can be confirmed from the FTR spectra. Some extra peaks can be observed for PVDF HFP gel electrolyte, and it is due to the presence of the organic solvent as well as the liquid electrolyte present in the network of PVDF HFP polymer. From the limiting current potential graph, the limiting current density calculated was found to be 5.94, and by applying it in the equation J limiting, we obtained the diffusion coefficient as 2.04, which is less than the PVDF electrolyte. So from the electrochemical impedance spectra, the calculated conductivity was found 0.848, was also very less when compared to PVDF based gel electrolyte. And this study was performed for a period of 30 days by recording this LSV. Here in this case also, as days passes, the limiting current value as well as the diffusion coefficient degraded. So one of the drawback, I think, for gel electrolyte is this is the phenomenon known as syneresis. Upon keeping the gel electrolyte for a long time, the liquid, if it is not stable, the liquid electrolyte may ooze out of the polymer network. So that may be the reason for my gel electrolyte also. I could see some of the liquid electrolyte they ooze out of the polymer framework. And so the ionic species responsible for the conductivity was lost. The performance degraded. So this is how the ESSC was fabricated in our lab for photo anode. The TiO2 was Dr. bladed on the FTO, and the area was confined to 0.25 centimeters square. Thus, it was then sensitized by dipping it into a solution of N1,9 for 12 hours, and we obtained photo anode. Counter electrode preparation was discussed before, and in between this photo anode and counter electrode, the gel electrolyte was sandwiched, and we obtained QS-DSSE. QS-DSSE is quasi-solid state. So the photovoltaic performance of PVDF QS-DSSE and the corresponding DSSE was recorded. Here is the electrolyte composition given, and from the graph, it's clear that the current was found to be 8.4 for liquid electrolyte based DSSE, and for QS-DSSE, that is PVDF based DSSE, is 5.03. So the efficiency also degraded from 3.55 to 2.28. 3.55 is for DSSE and 2.28 is for the gel incorporated DSSE. So for PVDF HFP based system also, the photovoltaic performance was carried out. The efficiency was found to be 3.85 for liquid electrolyte based DSSE and 1.91 for gel electrolyte based DSSE, that is QS-DSSE. So the current was found to be
please conclude. So 30 minutes, please
found to be less for QS-DSSE than the corresponding DSSE. So this is the time-dependent stability study of the device performed for a period of 15 days. Here the better performance was, in the previous slide we know that PVDF-based device shows the better performance compared to PVDF. So PVDF-based device stability study was performed and compared with the corresponding DSSE. So we can see that from the first two values, the degradation percentage was higher for DSSE. And when comparing the performance after 15 days, both DSSE as well as QS-DSSE show same kind of, almost the same percentage of degradation. So summing up, the performance of both gel electrolyte degraded as days passes, and the percentage degradation for PVDF is 55.87 and 48.03 after 30 days. The DSSC and QS DSSC was fabricated, and for PVDF based DSSC it's 2.28, and for the corresponding DSSC it's 3.55. For PVDF HFP based QS DSSC it's 1.91, and for the corresponding DSSC it's 3.85. The performance. So when comparing the performance of QS DSSC, PVDF shows higher efficiency than PVDF HFP based devices. The percentage degradation of both the devices, DSSC and DSSC, after 15 days, it almost completely degrades. So some of my research contribution in this area, that is a synthesis of jelly electrolyte and its application in DSSC by using some other polymers are listed here. And this is my acknowledgement to Dr. Sindhu Swaminathan, HOD Department of Nanoscience and Technology, University of Calicut, Sheetal MJ, MTech student, University of Calicut. Thank you.
Dear presenters, I'm saying once again, please stick to time. Okay, because we are having 27 presentations today. Please stick to time, maximum 10 minutes. If you have any questions, you can type in chat box. Please stick to 10 minutes. We are having total 27 questions. If you take each one 15 minutes, it goes to 9 o'clock. Okay, it's very difficult. Okay, please stick to time. Based on that, you can present. Okay. Yeah, next presenter. OP11, Santosh.
Yes, sir.
Yeah, you can start.
Am I audible, sir? Hello?
You are audible.
Okay, thank you. My name is Santosh Manopantar. I am pursuing my PhD in Visvesvaraya Technological University. And today I am going to present my presentation, that is preparation of colloidal silver nanoparticles. So in the introduction part, let me explain you first nanoscience, nanotechnology, and nanomaterials. So nanoscience is the study of nanomaterials, their properties and related phenomena. Or we can also say the rough definition of nanoscience could be anything has at least one dimension less than 100 nanometer. Nanostructured substances and nanosystems are fabricated and absolutely characterized with the aid of nanotechnological equipments and strategies. So next, nanotechnology is the application to produce devices and products. Nanomaterials are defined as materials having sizes in between 1 to 100 nanometer. Very next question comes, why we have to study nanomaterials? People are so much interested in studying the nanomaterials because of these properties. Let me explain you. At the nanometer scale, materials properties are different as compared to the bulk phase. Nanostructured materials are size and surface dependent properties. As a result of there is a change in the surface to volume ratio, widening of energy levels, and change in the density of charges. Therefore, the materials properties such as physical and chemicals are different at nanoscale compared to bulk phase. Next one is classification of nanomaterials. These nanomaterials are classified into 4 main categories, like zero-dimensional, they are called spheres and clusters. In one-dimensional, they are called nanofibers, wires, and rods. I have shown you the diagrams. Let me show you. So here I can show you. These are the nanofibers, wires, and rods. And 2D, that is in 2D, films, plates, and networks, you can see here. Sorry. And in 3D, nanomaterials, I have shown you here. Nanomaterials have all the 3 dimensions in the nanometer range, and the plates present only 1 dimension below 100 nanometer. Next one is applications of nanomaterials. These nanomaterials are used in the variety of fields. So let me explain. I have chosen some important applications. Here, these nanomaterials are used in the energy conversion, like fuel cells, solar cells, photovoltaic devices. In energy generation, if you say triboelectric, thermoelectric, piezoelectric. In piezoelectric, zinc oxide nanomaterials are used. Similarly, energy storage, supercapacitors. Nowadays, these supercapacitors and batteries are very booming field. Similarly, energy transmission like superconductivity. In energy saving, nano lubricants are also used. So like this, nanomaterials having variety of applications. So next one is metal nanoparticles. Sorry? Sorry?
Nothing. Continue, please.
Okay. Over the past few decades, researchers have become increasingly engrossed in the study of metal oxides. Metal oxides are ionic compounds that are made up of positive metallic and negative oxygen ions. The electrostatic interactions between the positive metallic and negative oxygen ions result in the firm and solid ionic bonds. Their D shells may not be completely filled, giving them a variety of unique properties that make them potentially of great use in electronic devices. These unique properties include wide band gaps, high dielectric constants, reactive electronic transitions, and good electrical, optical, and electrochromic characteristics, as well as superconductivity. Therefore, metal oxides are considered to be some of the most fascinating functional materials and have been widely exploited in various technological applications. These metal oxides play a very important role in many areas of chemistry, physics, and material science. The metal elements are able to form a large diversity of oxide compounds. These can adopt a vast number of structural geometries with an electronic structure that can exhibit metallic, semiconductor, or insulator character. In technological applications, oxides are used in the fabrication of microelectronic circuits, sensors, piezoelectric devices, fuel cells, coatings for the passivation of surfaces against corrosion, and as catalysts. In the emerging field of nanotechnology, a goal is to make nanostructures or nanoarrays with special properties with respect to those of bulk or single particle species. Oxide nanoparticles can exhibit unique physical and chemical properties due to their limited size and high density of corner or edge surface sites. So next, again, the question comes, where these nanoparticles are used. Significance of metal nanoparticles. These metal nanoparticles can be used in bioengineering, semiconductors, food packaging, electronics, catalysts. Like this, many application is there. And also these metallic nanoparticles like gold nanoparticles, silver nanoparticles can be used in the therapeutic application like cancer therapy and others, drug delivery. So many applications are there for metal nanoparticles. So next, in this, we have prepared silver nanoparticles. Let me tell you about the silver nanoparticles. Silver nanoparticles are increasingly used in various fields, including medical, food, healthcare, consumer, and industrial purposes, due to their unique physical and chemical properties. The unique size and shape-dependent optical catalysis and antimicrobial properties of silver nanoparticles have prompted increasing interest of chemists, physicists, and material scientists. Silver nanoparticles have attracted intense research interest because of their important applications in antimicrobial catalysis and surface-enhanced Raman scattering. Recently, silver nanoparticles have been frequently used in many textiles, keyboards, wound dressings, and biomedical devices. In the past, silver nanoparticles have attracted considerable attention due to the uncommon attributes of silver nanoparticles. They are used in different fields such as biomedical, in biomedical specifically, fast diagnosis, imaging, tissue regeneration, drug delivery, development of new medical products like that. Next, textile industries, food packaging, cosmetic industries, like that. These silver nanoparticles are used in many fields. Also, due to their remarkable protection against a wide scope microorganisms and medicinal properties, silver nanoparticles are utilized as anti-infecting agents, tranquilizers, conveyance agents, water treatment, forming, etc. Furthermore, due to their high conductivity, silver nanoparticles have found applications in electronic devices, inks, adhesives, facets, etc. Pastes, etc.
Please conclude, sir.
Sorry?
Please conclude. Thank you, sir.
Okay. Applications of silver nanoparticles. So now specifically I'm telling you the applications of silver nanoparticles. These silver nanoparticles are used in photo imaging, cosmetics and medical applications, biomedical, catalysis, food packaging and food safety, photothermal therapy, antimicrobial, various types of human cancer. So like that, many applications are there. So that we have chosen silver nanoparticles. So to synthesize silver nanoparticles or any nanoparticles, there are many methods like sol gel process, microwave assisted method,
Mr.
or
Santosh, please
precipitation method.
conclude your talk.
Please
Okay,
conclude.
okay.
Please conclude.
Okay, madam. Okay. So here we have used aqueous route or open air method. We have used capping agent as a chitosan. So this is our synthesis process. We have used two steps. In the first step, we have prepared a stock solution. In the second step, we have prepared colloidal silver nanoparticles. So these are the stable colloidal nanoparticles. First image shows as prepared colloidal. So this is after 3 months. And this is the UV image. And this is the fluorescence measurements. The size of the nanoparticles has been calculated by Debye-Scherrer formula, and we obtain the size as 20 to 25 nanometer. So for this, I conclude my presentation by these following points. Colloidal silver nanoparticles were successfully synthesized by chemical route using chitosan as capping molecules. As prepared, silver nanoparticles are highly stable and fluorescent. Confirmation of different size silver nanoparticles by optical absorption measurements. X-ray diffraction measurements reveals the crystallinity and phase of silver nanoparticles as 20 to 25 nanometer with face-centered cubic lattice structure we found. These stable colloidal silver nanoparticles used for a wide variety of biomedical applications, antibacterial agents, many applications. For many applications, these colloidal nanoparticles can be used. Thank you, sir. Thank you very much.
Thank you for your presentation. Yeah, next presentation, OP12, Dr. Rama Priyati, OP12.
Good afternoon, sir. This is Dr. Rama, Assistant Professor, Forensic Science Unit, Department of Chemistry, University College of Science, Osmania University. Here I am going to present my research paper entitled Extracellular Synthesis of Mono and Bimetallic Nanocomposites from Novel Strains of Purple Non-Sulfur Bacteria and Evaluation of their Biomedical Applications. Nanotechnology, which is an emerging fast-developing area with various applications in various fields of science and technology. So nanoparticles are nothing but the nano-sized particles which has a tremendous chemical, physical and optical properties. The usage of these nanoparticles plays an important role in agriculture and medicine, not only in agriculture and medicine, it also plays a very important role in arts, etc. Application of these nanoparticles in medicine solves many problems as these nanoparticles can be used for the diagnosis and treatment of various diseases. The main aim and objective of this particular research paper is synthesis and characterization of mono and bimetallic nanoparticles from a bacterial strain, that is a purple non-sulfur bacteria, and comparison of the applications of two bacteria. So synthesis of monometallic nanoparticles. Here, to a bacterial culture, extracellular synthesis, to the bacterial culture, we have added the metal concentration, 1 molar metal concentration. The three metals were selected here, that is the silver nitrate, gold chloride, and platinum chloride were selected. And to the extract, we have added the metal solution and incubated the solution, incubated the reaction mixture overnight. And coming to the bimetallic nanoparticles, to the culture, 1 molar concentration of metal 1 and 1 molar concentration of metal 2 were added, and the reaction mixture were incubated overnight. And after overnight, we have observed the reduction of the metal ion and identified that based on the color change, we have identified that the nanoparticles have been synthesized, and the synthesized nanoparticles were analyzed for the presence of the metal in the powdered sample. So after the synthesis of this nanoparticles, we have centrifuged it and we have collected the nanoparticles that were synthesized in this particular solution. Whatever the nanoparticles that were synthesized and centrifuged, those samples were dried and was given for the characterization. So coming to the EDX analysis, here it was done to find out whether the particular sample really contains only that particular metal or not. So here in this EDX analysis, we have identified that all the samples contains monometallic, contains a silver, gold, and platinum, whereas coming to the bimetallic, we have identified that the two metals were present in the sample, means the two metals were coupling with the biological components, the phytochemicals that is present in the bacterial sample. And DLS analysis. This analysis were done to find out the particle size of the nanoparticles, both mono and bimetallic nanoparticles, as well as the stability of the nanoparticles by analyzing the zeta potential. So here coming to the particle size, it was ranging from 33 nanometers to 220, 270, and more than 1,385 nanometers for silver, platinum nanoparticles. Whereas coming to the zeta potential, here the zeta potential values labeled or given as a red color indicates that those nanoparticles are less stable, and labeled as black color and indicated by a black color are moderately stable, and the nanoparticles, silver nanoparticles synthesized by both bacteria, have more stability. And antibacterial activity. So comparing to the two bacteria here, the antibacterial activity was done by using zone inhibition method. Based on the observation, here we have identified that the nanoparticles synthesized by using the strain 1, that is a Rhodopseudomonas palustris KRPR01, has less antibacterial activity, whereas coming to the strain 2, antioxidant activity, and here the antioxidant activity was done by using DPPH assay. So here also, whenever we are comparing with the first bacterial strain, the second bacterial strain has more antioxidant activity. And if we compare the antioxidant values with the control ascorbic acid, the second bacteria, the silver nanoparticle synthesized by using the second bacteria is showing almost a similar amount of the antioxidant activity when compared to the ascorbic acid, and hemolysis assay. Here in case of hemolysis assay also, whenever we are comparing the both first bacteria and second bacteria, here the second bacteria is showing the highest hemolytic activity. And anti-cancer studies. Anti-cancer studies were done by using a MTT assay. So here the antioxidant studies were done on 4 different cell lines. That is HeLa cell lines, DU145, MCF7 and Miyabaka 2 cell lines. On comparing the cytotoxic activity in all the 4 cell lines, the second bacteria, the nanoparticle synthesized with the second bacteria is showing the more cytotoxic activity when comparing to the first bacteria. Coming to the conclusion, here in this bacteria, the 2 bacterial strains has the capacity to reduce the metal ions and convert them into nano-sized particles. Based on the characterization, we have identified that the second bacteria is reducing the metal ion very much when compared to the first bacteria, where the synthesized nanoparticles have the size of less than 200 nanometers. Whereas coming to the first bacteria, in case of silver and platinum nanoparticles, it is showing the more size, that is above 1000 nanometers. So we can say that the second bacteria has more efficiency to reduce the metal ions, and based on the applications, the antibacterial activity, antioxidant, as well as the hemolytic and cytotoxic activity, when comparing to the first bacteria, second bacteria is showing the higher efficiency. Therefore, we can say that the nanoparticles synthesized by using the second bacteria can be used in various fields of medicine and biology. So in case of medicine, we can use these nanoparticles for the diagnosis of various diseases such as cancers, etc. And also the nanoparticles nowadays can be used in various treatments. So future, as the size of the nanoparticles is very large, various optimization studies need to be done in future to reduce the particle size of the synthesized nanoparticles and also to increase the stability of the nanoparticles, because here most of them are moderately stable and few are less stable. Only 1 or 2 nanoparticles are having the higher stability. So the optimization studies are required to increase the stability of the nanoparticles and also to improve the applications in various areas such as biology, medicine, arts, agriculture, and industry. I acknowledge my supervisor, Professor M.B. Pratap Rudra, Department of Biochemistry, Osmania University, for his guidance and support in my PhD research work. And I also acknowledge the Department of Chemistry, Department of Biochemistry, DST PERS, Central Facilities and Research Department, Development, Osmania University College of Technology, Osmania University, and IICT Tanaka, Hyderabad, for providing the necessary infrastructural facilities. And I also acknowledge UGC for supporting the financial assistance for carrying out my research work. Thank you.
Yeah, thank you for your presentation. Yeah. Next presentation, OP13, Anupama Anjali. Next presenter, OP13.
Yes, sir. Am I audible?
You can share your screen.
Hello?
Yes, sir. Good evening. Good afternoon, everyone. I'd like to
Kindly come to your work, your own work. No need to present your introduction. Kindly come to your results.
Okay, ma'am. Welcome to my presentation on green catalyst, environmental to combat environmental problems. This is the overview of my presentation. It includes introduction about catalysis, and then I'll go to my in brief about the introduction. Chemistry is the most demanding topic to overcome the environmental issues like toxic waste, cost, and energy, which is a great challenge for the chemists. The way we are dealing with such issues are inappropriate to minimize the contamination to air, water, and land. Green catalysts are those chemicals which can be utilized as a catalyst, and they cause either no harm to the environment or have minimal effects on the environment. There is a rising demand for more eco-friendly processes in the chemical industry. Many articles have been published in the last 2 decades on green chemistry and green catalysis. In recent years, green chemistry has attracted the attention from the synthetic community. So here I will give a brief view for the efficient catalysis. About catalyst. A catalyst is a substance that increases the rate of a reaction or speeds up its rate by decreasing the activation energy. These are the different types of catalyst, which includes positive catalyst, negative catalyst, auto catalyst, and induced catalyst. Green catalyst. All such catalysts which can be recycled, reused, cost-effective, energy-efficient, such catalysts come under this category. They may be solid-supported or metals which possesses magnetic nature, nanoparticles at atomic stage which can be catalyzed efficiently, and ionic liquids which can serve as solvents and catalysts are the examples of such chemicals. These are some of the principles of green catalysts, which are magnetic in nature, mainly solid supported, easy to remove, maybe at atomic scale, cost effective, recyclable, and maybe in liquid form or can be as solvent. So the catalyst can be supported on the solids such as clay, alumina, cellulose, polymer resins, solids, magnetic metal, silica, and monomer. So silica supported catalyst for green catalysis. Silica supported catalyst is one of the promising catalysts carrying out potential reaction selectivity. It is bound to have strong hydrogen in the substrate and the catalyst surface.
I think the participant got disconnected on her own.
Hello.
Yeah. We request Mr. Vijay Singh to stop for a minute. Yeah. Miss Anjali, kindly just go to your result slide and present. Only your result slide. Only your result slide, please.
Okay, ma'am. Sorry for the interruption.
Show only your result that you have got.
Yes, ma'am. So this one is the data which I have collected and get the information. Upon silica gel, chlorosulfonic acid is bound and it gets absorbed. Then the absorbed chlorosulfonic acid is used in the reaction as a catalyst. Later on, it is filtered, then separated, then again the second reaction is done and workup performed. So in this way, the catalyst is used multiple times. If solid support used for catalyst binding. The solid phase catalyst was found recyclable and its efficacy was tested by recycling them for conducting experiments. Same amount of silica during workup lost. So the same efficiency of catalyst was not achieved. Some quantity of catalyst reused 5 to 6 times and got sufficient amount of product. So the catalyst can be recycled, reused, which is a part of green methodology. So this is the mechanism of pigeonelli reaction. It's a multi-component reaction of cyclohexanone and thiourea and aldehyde. And the silica sulfonic acid releases H plus ions during the reaction, which reacts with cyclohexanone, which further on proceeding in the final step, and then the condensation occurs to form the fuse-bringing compound quinazolines. The compound prepared in lab compliance with this data. Data also detect this compound, which has the molecular formula C14H15Br N2S. This is the data for the, we have put 3 data here. One is the 13 carbon, next is the proton NMR and the mass data. In the 13 carbon, we have found 14 carbon peak shown proton data also in compliance, and the mass data also indicates comes at 325, which compliances with the molecular weight at 322, and bromine has 2 isotopes, so 2 peaks are observed here. The fuse, finally coming to the output, the fuse ring quinazolines are biologically active actually. Several activities of it, that is antifungal, antibacterial, have been reported. And MCR reaction number of steps have been reduced, taking silica sulfonic acid leading to quinazolines. It's a 100% green methodology because the product formed favors biologically. It is a solvent-free, neat reaction, solid supported, recyclable, reusable catalyst. So here we have covered the aspects of green chemistry. And I would like to acknowledge my PhD supervisor, Dr. Nisha Saxena, for helping me and guiding me in this presentation. Thank you.
Thank you for your presentation, Ankuma.
Thank you, sir.
Next presenter, OP14 Vijay Singh. Sir, you can start your presentation. Vijay Singh. OP14 Vijay Singh.
Mr. Vijay, please start your presentation.
Hello, my voice is clear, madam? No?
Yes, yes, yes.
Okay.
Continue.
Good evening to all of you. Myself is Vijay Singh. I am working in the chemical engineering department in IIT Lucknow. My presentation topic is citrus fruit juice clarification by PET column and membrane coupled operation. Actually, I want to treat the PET column used for pre-treatment of juice. After that, some impurities is there that can be removed by the membrane operation. That's why I use for combined operation. The whole overall presentation is divided into the following section. First is introduction, second is material and method, third is representation, after that conclusion and references. Introduction for phytozoos, especially I have taken for cider in phytozoos fruit, especially I have taken Mosami. It is seasonally available. After that season, after when in particular time that Mosami is available, after that that is not available. In citrus fruits, it's a rich source of vitamin C content and other things that can be a good antioxidant property. And membrane separation and evaporation operation a lot of operation is there, and memory separation evaporation are used for clarification used before that. And to reduce the falling, improve the if we want to the store for a juice for a long time, they can just first you preheat the juice, then after that you just go to the other side. And a lot of common method is there for such as the sanctification, binding agent, and other enzyme treatment. But some others are they all are very costly. That's why I go to the some other new pre-treatment at a PET column. Okay. So in material and first I take the fresh Mossambi juice and screw type extractor that can be purchased from the market, local market quantity. And for clarification purpose, I use the polyamide membrane. It's a particle size 2.5 micrometer under the micro, which is come in the under the microfiltration range. These are my systematic scheme for whatever thing. Initially, first I have to go to the pre-treated and setting of sorting of the Mosami from the local market. After that, we'll go to the washing and just cut into halves, and then after that go to the pre-treatment. In pre-treatment, first we go to the the coupled operation. First go to the pad column operation. After that, whatever output come, that can be collected in separate bit and go to the membrane separation operation. And for packing for in packed column, two types of packing material I have taken. First is glass bead, other is the and other is the ceramic, and other one and last one I have used for combined operation. That combined operation, first we go to the two packed column I have arranged in series. First is the first column I filled with glass bead material, and other is that whatever output come, that go to the second column, that second column also filled with the ceramic bead. After that, just whatever output will come, that can be go to the membrane separation process. Operation in membrane sorter, that is my experimental setup. Here they compare for pressure generator. For I have used the compressed air cylinder, and this is my membrane cell filtration cell. It's here it's a diameter point 5 mm, and that is it's a cylindrical cell, and here I with the help of compression I generate the pressure will generate, and pressure near about low pressure I have used for the for clarification for juice. Near about 69 kPa Pascal kPa I have used for generate the pressure. These are my experiments. A lot of experiment I have conducted, but here I have shown for packed juice packed juice column with during the packed juice whatever I have set up, I have used for different flow rate. I have used for conduct the 0.39 to 1.3 for same for different. After that, I just observed that a let low flow rate, the quality the pretreated juice quality has more compared to the raw juice. And after that, clarification purpose for 69 kPa Pascal I have used, and polyamide membrane I have for 2.5 microfiltration membrane I have used for clarification purpose. These are the following properties I have do for analysis for raw and pre-treated and clarified juice. First is Brownian index, other is clarity. I have measured pH. Basic that show be how much your solution is acidic or pH. Acidity I have measured, viscosity I have measured, density with the help of cyclometer bottle I have used, pectin content I have pectin content measured with the help of alcohol with the help of methyl alcohol. At the last I have soluble solid in terms of a big degree of bricks that can be measured by the refractometers. After that, when conduct the experiment, after that the clarified juice I just pass through the microfiltration membrane. Here we observe that the combined operation have juice quality is more than, and that combined juice quality if you go to the glass bead packing material we use, then the flux will be immediately sharply decline. After that, the ceramic bead is better, more better than the compared to the glass bead. If we go to the combined operation, that is quality, that is its permeate flux is also very good compared to the both single and double. So that's why we use for combined operation. Combined operation at multiple at single at low flow rate. We just use for. After that, we just go to the measure the alcohol physical chemical properties of Mosam juice for pre-treated juice and clarified juice and raw juice. So here we have observed that the maximum clarified juice the alcohol insoluble solid in 95% alcohol insoluble solid are removed, and that the clarity will increase 0.65 to 98.4%. Raw juice clarity is 0.65 and alcohol insoluble, and then after that clarified juice is 98.4. For single column it is also very low. For with glass bead 5.2, and for ceramic bead for 12.30. After that combined this is 16.3. So then finally go to the clarification, for its quality will also very high, and the viscosity is also very viscosity also removed for combined operation 75.4. The single raw juice viscosity I have whatever I have taken, it's 100%. After that for single we use the glass bead with 25.13% viscosity will reduce, and the single with glass bead, and for ceramic bead 35.45, and for combined 42.32% viscosity has reduced. So clarified juice quality has more compared. That can, if on the basis of this table we can use for that clarified juice can be stored for a long time for a particular operating condition, not for you can use for anywhere for a condition. After that, the following conclusion I have observed: single pad column pretreatment is not good enough to remove high molecular weight compound. If you use a single column, suppose if you go to the single for glass bead and go to the simple for ceramic bead, that is not enough. In column pretreatment, juice quality depend on the whatever if we go to the packing, that all the juice pretreated juice quality depend upon the packing material. If we go to the low dense packing material, that quality is more. That high dense packing material, its quality also definitely will change. In combined column operation has maximum ability to remove the impurities. If we go to the in terms of alcohol insoluble solid, AIS, whatever viscosity reduction and certain other properties. Suppose in the clarity, in the clarified juice can be stored for different if we use that clarified juice at low temperature, that can be stored for a certain long time period for different operating conditions. At the last, whatever I have references I have taken, I just mention it, and at the last, I thank you. Thank you for your.
Okay, thank you, Vijay, for your presentation. Yeah, next presenter, OP16, Dr. Reena.
Thank you, sir.
Yeah. Next presenter, the OP16, Dr. Reena.
Am I audible?
Yeah, Adi, you can start the presentation.
Good evening to all. Myself Reena Prihar. I am research scholar under the supervision of Dr. N.K. Singh from the Department of Chemistry, University of Lucknow, Lucknow, India. The topic of my presentation is oxygen evaluation electrocatalytic properties of SM1-X, SRX, NiO3 obtained by malic acid sol-gel rule. First one is the introduction. At the present, fossil fuels are the main source of energy consumption, but these fuels are limited, non-renewable, and causes environmental problem due to the unburned CO, CO2, SO2, and NO2. So researchers have paid great attention to develop the low-cost, renewable, and eco-friendly energy sources. Oxygen evaluation reaction is an important process in energy storage and conversion, especially the water electrolysis. Literature survey reveals that the transition metal mixed oxide with spinel and perovskite structure have been used as the electrocatalyst for the oxygen evaluation reaction. However, the samarium-based perovskite oxide have been very little investigated towards the oxygen evaluation reaction. Therefore, in the present work, samarium nucleates have been synthesized by using the malic acid salt gel route, and their physicochemical and electrochemical properties have been studied with regards to the oxygen evolution reaction. Synthesis. Samarium nucleates were synthesized by using the malic acid salt gel route, which is described here. Firstly, the stoichiometric amounts of the metal nitrates were dissolved in the distilled water, and an excess amount of malic acid was added. The pH of the starting solution was adjusted at 3.5 by adding the ammonia solution. Then the mixture was concentrated over the water bath, and a gel-like mass was formed, which was further decomposed and sintered at 650 degrees centigrade for the 5 hours to get the desired oxide materials. These are the oxide materials prepared by the malic acid salt gel route. Physicochemical characterization. The oxide powders were characterized physicochemically by using the scanning electron micrograph and the XRD. First one is the SAM. Figure 1 shows the SAM images of the oxides samarium SM.8 SR.2 NiO3 and SM.2 SR.8 NiO3 at the magnification 200. And from the figure, it is clear that the texture of the oxide powders appears similar, and the flakes-like structure observed in the micrograph. Second one is the XRD. Here the figure second shows the XRD patterns of the S powders, oxide powders SM.2 SR.8 NiO3 and SM.6 SR.4 NiO3 sintered at 600 degrees centigrade for the 5 hours. The previous cut phase of the oxide powders was confirmed by recording the XRD, and the crystallite size was found to be approx 30 nanometer for the SM.2 SR.8 NiO3 and 40 nanometer for the SM.6 SR.4 NiO3 oxide by using the device curve formula, which is given by S is equals to 0.9 lambda by B cos theta. Electrochemical characterization. For the electrochemical characterization, oxide powders was transformed in the form of oxide film electrode on the pre-treated nickel support by using an oxide slurry painting technique. This figure shows the experimental setup for the electrochemical characterization, and all the electrochemical experiments such as CV and cyclic voltammetry and anodic tuffle polarization have been performed in the 3 electrode system. Oxide material was used as the working electrode, and HgO 1 molar KOH was used as the reference electrode, and platinum foil was used as the counter electrode. Cyclic voltammetry CV. Here figure 3 represents the cyclic voltammogram of the oxide powders on the nickel support in 1 molar KOH at 25 degrees centigrade. Each voltagram shows the anodic and cathodic peaks prior to the oxygen evolution reaction, and the values of the cyclic voltammetric parameters were estimated from this CV curve, and these values are given here in the table 1. The variation of the scan rates on the redox behavior is also studied in the 1 molar KOH at 25 degrees centigrade. Here figure 4 shows the CV curve of the oxide film electrode SM.2 SR.8 NiO3 at the different scan rates. From the figure, it is clear that as the scan rate increases from 20 to 120 millivolt per second, the anodic peak shifted toward the higher potential, and cathodic peak shifted towards the lower potential. And this figure 5 shows the plot of mod JP versus the under root of scan rate, and figure 6 this shows the plot of Q versus 1 by under root of scan rates. And anodic polarization curve tuffle plot. Figure 7 shows the tuffle plots for the oxide powders in the 1 molar KOH, and the electrokinetic parameters were estimated from here, figure 7, and these are values are given in the table 2. An order of reaction for the order of oxygen evolution reaction was determined by recording the anodic polarization curves in the different KH concentrations at 25 degrees centigrades. And then the plot, figure 9 shows the plot of log J versus log OH minus, and the slope of this plot gives the order of the reaction. And thermodynamic parameters, the effect of the temperature on oxygen evolution reaction was also studied. And here figure 10 shows the anodic polarization curve for the SMNI3 film electrode at the different temperatures, 20 degree, 30 degree, 40 degree, and 50 degree centigrade. And figure 11 shows the RNS plot at the potential 660 millivolt for the oxide electrodes, and the thermodynamic parameters values are given in the table 3. Conclusion, the previous CAT phase of the oxides were confirmed by using the XRD, and the pair of redox peaks was observed in the cyclic voltammogram of each oxide electrode. Or the oxide SM.2 SR.8 NiO3 was observed to be most active with the current density 4.1 milliampere centimeter inverse 2 at 800 millivolt. Thank you.
Thank you for your presentation, Reena. If you have any questions, you can type in. You can write in your chat box. Okay. Now we can move to next presentation. OP 17, P. Chandrakant Singh. P. Chandrakant Singh, OP 17. Is he there? Chandrakant Singh.
Sir, am I audible?
Yeah, audible. Yeah, you can start, sir.
हेलो, हाँ देखा था क्या? हाँ हाँ हाँ.
Good afternoon, and I am P. Chandrakant Singh, the PhD scholar, Department of Physics from KIT deemed to be University of Bhubaneswar. Here I am going to present the topic USB digital microscope endoscope camera, a good tool for quick morphological characterization of laser induced microstructures, silicon. This is my outline of initial the introduction, then the limitation, objectives, then go for the experimental technique, results, conclusion, then acknowledgement and reference. So generally the texturization can be carried out by the different methods, at the wet etching method or the dry etching method. And but in this case, I have used the dry etching method by using the laser texturization, which is the very vital technique, having the distinct advantages like well controllable, highly reproducible, and it is a single step processing method and dry process and eco-friendly. However, in the texturization work, for doing it effectively, we must have initially go for the morphological characterization of the generated structures and to know the dimension of the laser damage spot site, so that after confirmation we can go for the brief morphological characterization by using the electronic scanning microscope. And generally, already I have told you that texturization is two types. One is wet etching method, another is dry etching method. However, these methods have some limitation, that is difficult to control, slow growth rate, relatively high cost, and not reproducibility. So another thing is, for the characterization, generally we go for the morphological characterization, we go for the optical microscope or the electronic microscope, which is very costly and bulky. So before going to the detailed morphological characterization, here I have demonstrated the USB digital microscope endoscope camera, which is used for the quick morphological characterization. After the texturization by using a laser, initially I have to know whether the texturization or the structure is grown or not. That is my first task. So for knowing this one, I now need to go for the scanning electron microscope or the optical microscope, which is very costly and bulky. Instead of that, I can use this UDME camera. Secondly, I can measure the dimension of the damage spot, and by getting the damage spot dimension, I can change the laser parameter, processing parameter. And this UDME camera is compact in size, portable, and easy to handle, and inexpensive for this task. And for the texturization, I have used the nitrogen gas nanosecond laser, and whose wavelength is 337 nanometer, and the pulse energy is 170 microjoule, and the average power is very low, that is 3 milliwatt, which is very difficult to modify the surface of the silicon, what I have asked first time in India. And its beam size is 3 cross 7 millimeter. This is the laser workstation, what I have indigenously developed in our lab, photonics lab in KIT University, and it is also patented by the NRDC, Government of India. And my task, the second task was for the morphological characterization. For the quick morphological characterization, I have used the USB digital microscope endoscope camera. And for the detailed morphological characterization, I have used for FESM, fields emission scanning electron microscope, and I correlated with the own arranged USB digital microscope endoscope camera. This is the camera whose cost is very low. And for the initial stage, for the quick morphological characterization, for knowing the surface texturization, we can opt this low cost UDME camera. This is available in the market, and you have to do some arrangement for the X and Y as well as the for Z direction for focusing onto the sample. And this camera is calibrated by imager software, and it is very important for deciding the working parameters of the generated microstructure. Now this is my results and decision or discussion. In case of results and discussion, initially I have demonstrated the overlapping of the laser damage spot. While I am going for the texturing the surface, initially I have to measure the damage spot of the laser damage spot area. Secondly, I have to go for the overlapping of the laser damage spot in vertical direction. Then I go for the large area texturization, where I have used the raster scanning mode. So this is the D diameter of the laser spot, or if you take the rectangular shape of the what is the damage spot, then we can take it is W, the width of the damage spot, and this is the gap between two damage spot at specific velocity, scanning velocity, and this is the distance displacement of the damage spot from the initial point to the next, the initial point of the another damage spot, and this is the overlapping area. And this overlapping laser damage spot overlaying can be calculated by in the percentage way. And after getting the vertical overlapping, we go for the horizontal overlapping. This is the horizontal overlapping damage spot, and the percentage can be calculated by D minus L by D into 100. That is the percentage. So after texturization, I have textured the silicon by using nitrogen nanosecond laser at repetition rate of 20 Hertz. Pulse duration is of 0.3 nanosecond, and the scanning velocity is of 0.5 millimeter per second. And this imaging is taken by using the UDME camera, USB digital microscope endoscope camera, which is the very low cost camera. And the length of this structure is of 500 micron. By capturing this image by using the UDME camera, I can easily measure the damage spot and the width and the separation between the conjugative laser damage spot, okay, which is very important for me for getting the vertical overlapping of the laser damage spot for generating the large area texturization. And this is the FESCM image of the same structure, and there also I get the similar result, which is captured by using the UDME camera. This is the damage spot area enclosed by the rectangular shape, and this is the unstructured area, and the rectangular space, the damage spot is separated by the double-sided arrow line. And for the vertical and the horizontal overlapping, I have done some laser processing parameters and the mathematical calculation while changing the velocity of the scanning velocity at constant repetition rate. I have only changed the scanning velocity of the sample at initially 0.5 millimeter per second. I could not get any overlap percentage. Then I reduced the scanning velocity to 0.4, still not getting. Then at 0.3, then I got some overlapping of the damage spot, that is 11.76%. Then when I reduced to the scanning velocity to 0.2 millimeter per second, I get the overlapping, that is of 41.717%. This is the optimized overlapping condition. When I reduce the scanning velocity to 0.2 millimeter per second at 20 Hertz repetition rate of the 337 nanosecond laser, then I observed by taking the imaging using the UDME camera, USB digital microscope endoscope camera, I observed that there is not having any untextured area. That means the laser spot damage is completely overlapped. Okay? So I decided by using these parameters, I can go for the large area texturization. And this is the FESCM image for confirming the UDME camera. Then I decided for the large area texturization, which is very important for application of the optoelectronics application like solar cell, okay, LED, and so on. This is the UDME camera images, and this is the FESM images, which is correlated to it. Now the conclusion: for the quick morphological characterization, anyone can own this low-cost UDME camera, and it is anytime you can use. It is very handy, and it is first time used in our lab. And by this camera is calibrated by using the image software, and it is easy to handle, inexpensive, and can be used for the quick morphological characterization. For this work, I acknowledged for the financial support to the SAB, Government of India, and this is my references. And thank you for your kind attention.
Thank you for the presentation. Next, we will move to next presentation, OP number 18. Paris, listen. Paris, listen.
Yes, sir.
Yeah, you can start. You can share your screen.
Yes. Good afternoon, everyone. I like to thank the chairperson, and also like to thank the organizing committee of ICSEM 2021 for giving me this opportunity to present a part of my work in this conference. And I'm Lisham Paris Jammu, research scholar in physics department, Manipur University. So, can you hear my audio, sir? Hello?
Yeah, audible. You can continue.
Oh, okay, sir. So I'll be talking on the effect of the substitution at NN site on the structural and electrical properties of the rare earth manganite, that is YMNO3. Let me give a brief description on what is multiferric materials and why is it important. So ferric is the material that adopts a spontaneous switchable internal alignment, whether it may be ferromagnetics, ferroelectrics, or ferroelastic. So multiferrics are a new group of material in which two or more than two ferrous order exist. So in ferromagnetics, there is alignment of electron spins, which can be switched by the magnetic field, and in ferroelastic, electric dipole moment alignment can be switched by the electric field. And lastly, in the ferroelastic, strength alignment can be switched by the stress field. So, what is the effect of the multiferrous materials that we are finding a new field, a great research interest in this group of materials? So, there exists a strong possibility of the magnetoelectric coupling in these materials, and they have many functionalities like in the field as an actuators, transducers, and storage devices, and multi-state memory devices. So, and overall, the magnetoelectric multiferrics are of has a great potential applications due to the linear coupling between the ferroelectric and the ferromagnetic properties. So in my sample, there is YMNO3. I have prepared the sample by Sol-Gel method, and this is a brief flow chart of how I prepared my samples. And this is the characterization. This is the characterization that I've done for my samples. There is the for the structural characterization, I have done the XRD, FSM, FTIR Raman. And for electrical properties, I have measured using the Agilent SP L-star meter. I have studied on the dielectric properties, AC conductivity, as well as the impedance analysis to study the relaxation behavior of the samples. And this is the XRD that are my samples. That is for the pure and the samples with chromium, and also for the product samples with chromium, nickel, and chromium and iron. So this is the XRD pattern for the pure YMNO3. It shows the formation of pure hexagonal phase without any impurity phase. But as I dope with the other transition elements, there is chromium, and for as well as for the product samples also, we have seen some of the orthorhombic peaks, which signifies the onset of to orthorhombic phase transition from hexagonal to orthorhombic phase transition. So we have seen here a few of the orthorhombic peaks as denoted by the asterisk marks, and the below the table is the latest parameters and the crystallized size of the samples. So here for the latest parameters for the dot sample, it seems to decrease, particularly for the dot sample that is with the chromium. Is slightly decreased than the pure YMNO3. This may be due to the slightly smaller radii of the chromium ion. But for the dot samples, there is chromium and nickel, and the chromium and iron The latest parameter is slightly increased, which lead to the increase in the cell volume, which may be due to the larger, slightly larger ion radii of the nickel and the iron as compared to the manganese ions. And this is the river refinement that I have done for the pure YMNO3. And this is the same micrograph of my samples. As it can be seen from this micrograph, there is no micro cracks. And from the grain size distribution, you can see that the grain size is very much larger than the crystallite size. This may be due to the growth of the particle as I sinter the particle at very high temperature, that is around 1100 degrees Celsius. And this is the FTIR studies. Here we can see the vibration peak for in all the samples. So the peak around 594, which may be due to the MNO stretching vibration, and 484 is due to the wire stretching vibration, and 420 is due to the OMNO bending vibrations. And here is the Raman analysis that are my samples. And from the Raman analysis result also, we can see how the orthorhombic phase transition is observed for the dog sample. As we know that according to the group theory, there are 16 to 60 phonon modes, among which 38 are Raman active. And for my synthesized samples, we have got the Raman active modes of A1, E1, E2. With the maximum intensity percentage of the A1 mode. This confirm the hexagonal phase formation of my pure sample as well as the dog samples. For the 4 dog samples, we have seen the emergence of this orthorhombic peaks, that is the B2Z, which is due to the joint symmetry oxygen stretching vibration mode. Yeah. So the Raman mode around A1 is due to the MNO apical stretching vibration, and the vibration mode at 130 is related to the yttrium ion stretching vibration. And from the Raman spectra, I have deconvoluted using Pico software, and here is the peak position of the Raman mode. But here I have observed that the Raman mode are slightly shifted to the higher value region. This may be due to the slight change in the lattice parameter, which will affect on the bonding of the MNO. This leads to the change in the position of the Raman phase. And here is the dielectric properties of my sample. I have measured the dielectric properties for the frequency variation at room temperature, as well as for different temperature variation, that is up to around 470 Kelvin from room temperature. And here the dielectric properties is observed to have the usual dispersive phenomenon, that is the high dielectric constant value at lower frequency. But as the frequency increases, the dielectric properties decreases. This may be due to the high and this is the dielectric loss data, the AC conductivity. And this is the impedance analysis to discuss on the relaxation phenomena of the material. And this is the real part of the impedance as a function of the frequency. And this is the imaginary part. And yeah, this is the Nyquist order for plot on our sample. And here we have seen the existence of the semicircle. So it shows the non-divided relaxation type of relaxation type in our behavior, and 2 semicircle is observed for the third sample, this dog with chromium and nickel. This is one of the peak at lower frequency is due to the blend boundaries, and higher frequency due to the blend. And it shows this asymmetric semicircular arc shows the non-divided relaxation type in my samples. And this conclusion, so I have synthesized the pure and the dog YMNO3, that is for the with the transition materials, and XRD shows the single hexagonal structure for YMNO3 with the orthorhombic phase transition is observed for the product sample. And efficient index is also studied. And from the dielectric property, it shows that charged conduction mechanism in my samples are primarily activated as seen from the temperature dependent dielectric status. And from the impedance spectroscopy reveals the relaxation that peaks for all the samples, and the nucleus flow shows that an undivided relaxation that is prevalent for all my samples. These are references. Thank you.
Thank you for the presentation, Lisa. If you have any questions, you can write in chat box. Okay. We will move to next presentation. That is OP 19, Vipin Kumar. Now we will move to next presentation, OP 19.
Good afternoon, sir.
Yeah, you can share, sir. You can share your
Okay,
screen.
sir. Okay, I am sharing.
Yes.
My presentation is visible?
No, not yet, sir. Not yet share.
Now it is visible?
Yeah, right. Now you can,
Okay, sir.
you can make a slideshow.
Good afternoon to one and all present here. Myself, Dr. Vipin Kumar from Kite Group of Institutions. The title of my presentation is Fabrication and Characterization of Sol Gel Screen Printed ZNCDO Thick Film for Optoelectronic and Photovoltaic Technologies. The co-authors are Renu Kumari, Dhiren Kumar Sharma, Sweta Shukla, Akanksha Agrawal, V.K. Sachang, and we all belong to Kite Group of Institutions, Delhi NCR, Ghaziabad. The outline of my presentation is introduction, experimental section, and characterization techniques, and conclusions. Among introduction, cadmium oxide is a significant degenerate n-type semiconducting material with initial crystal structure and having a large type optical band gap within the limit 2.2 to 2.7 electron volt. The structural, optical, and electrical properties of cadmium oxide films can be enhanced by doping of various metal oxides. Zinc is a transition element and it has good optical and electrical properties. Zinc is a good dopant to alter the optical and electrical properties of cadmium oxide films. Dead and doped cadmium oxide films have wide application in solar cells, optoelectronics, photodetectors, photovoltaic devices. This is the synthesis method. The material for gel formation were 95% of molecular weight of cadmium acetate dihydrate and 5% of molecular weight of zinc acetate dihydrate as a solute and ethylene glycol as a solvent. The prepared solution was stirred for 2 hours at 80 degrees on magnetic stirrer. Brownish gel was formed after 2 hours and then gel was kept over cooling for 2 days. The film was developed through the standard screen printing setup shown in figure. This is the screen printing setup for preparing the film from gel prepared by gel technique. Now we come to the properties. First of all, structural analysis by X-ray diffraction. The figure shows the X-ray diffraction of the prepared zinc 5% doped CDO films, which authenticates the polycrystalline nature and creation of pure cubic structure of CDO. The absence of extra peaks in the XRD confirms the ZN doping do not upset the lattice structure of CDO. This is the surface morphology. Densely, tightly packed tissue-like structure were observed. EDEX also confirms the presence of cadmium, oxygen, and zinc elements through the EDEX analysis, which confirms the doping of zinc in CDO. Now, optical analysis by UV visible spectroscopy. The optical band gap of film was evaluated by reflectance spectra shown in figure. The energy band gap of film is found to be 2.60 electron volt by plotting the graph between energy on x-axis and the h nu ln r max minus r minimum upon r minus r minimum, where r max is the maximum reflectance and r minimum is the minimum reflectance. R is the reflectance at any intermediate state. Now PL spectra. Through PL spectra, we observed that one strong peak were observed at 500 nanometer attributed to green emission. No peak related to UV and IR region were observed. The high intensity peak observed in photoluminescence spectra indicates that the transition is mostly from the conduction to valence band. This is the electrical analysis. The electrical study of Zn 5% doped CDU film was done by E3 electrometer. RNS plot for DC conductivity of Zn 5% doped CDU film depicts rise in conductivity with rise in temperature. The RNS plot confirms the semiconductor nature of the film. Now conclusion, 5% zinc doped cadmium oxide films were prepared by Soldier screen printing process. X-ray diffraction study supports the polycrystalline nature. SEM image confirms the impact of gadolinium doping on morphology of the film. Presence of cadmium, oxygen and zinc elements was confirmed by EDEX analysis. The optical energy band gap of developed films is found to be 2.60 electron volt. The electrical study affirms the semiconductor nature of the film. These observations indicate the suitability of zinc doped CDO films for electronic and photovoltaic technologies. Thank you.
Yeah, thank you very much for your presentation, sir. If any questions, you can write in chat box. Now we will move to next presentation. Next presentation, OP number 21, Komal.
Sir, Komal is not available in the meeting, sir.
Okay, now we will move to next presentation, OP 22,
Yes.
Kumar. OP 22, Kumar Shweta.
Hello.
Yeah, it's audible.
Good evening, everyone. My name is Kumar Shweta, and I am doing PhD under supervision of Professor K. Kumar at the Department of Physics, IIT ICSEM Dhanbad. So my topic is up and down conversion photoluminescence, a study of YF3 HOYV doped phosphor. So firstly, I will represent the outline that what I'm going to talk about. Firstly, introduction, then experimental synthesis method, then characterization and result, then conclusion, and then references. So introduction. So what is luminescence? Luminescence is a process of emission of photons, not due to thermal radiation. So it is a kind of cold body radiation. So different types of luminescence processes are there, like bioluminescence, chemiluminescence, photoluminescence, cathodoluminescence, and electroluminescence. So as its name inherits the processes which leads to the emission of photons, or we can say leading to the process of luminescence. In bioluminescence, biological phenomena are involved in chemiluminescence, chemical processes occurs due to which luminescence occurs, and photoluminescence, photon absorption takes place, and then luminescence occurs. Similarly, cathodoluminescence, electron impact caused luminescence are there, and electroluminescence is the cause of electrical region is region of luminescence is the electrical phenomena. So there is two types of photoluminescence: first fluorescence and second phosphorescence. So in fluorescence, the energy transition between singlet to singlet state, and in phosphorescence, it is from triplet to singlet state. So these are the basic difference in fluorescence and phosphorescence. So here I'm going to talk about upconversion cause of basically part of fluorescence. So upconversion is a process of conversion of lower energy photons into high energy photons. So basically it absorbs two or more lower energy photons and converts it to high energy photons. Similarly, downconversion is a reverse process in which high energy photons are converted into lower energy photons. So there are some other processes also involved which photon sensitized energy transfer of conversion, cooperative luminescence, energy transfer of conversion. Hello, am I audible? Hello.
Yes, you are audible. Please continue.
Okay. So there are other processes like energy transfer of conversion, excited state absorption, cooperative luminescence, So there are asymmetric representation of these processes like ground state absorption, in which atom in ground state absorbs a photon and goes to higher excited state. Then by some other process it goes to next excited state and then makes transition to give luminescence. Similarly, in excited absorption, a atom which is already in the excited state absorbs photon and makes transition to next higher state, and uh, then the transition from that higher state gives the luminescence. So energy transfer of conversion and sensitized energy transfer of conversion is basically the same process, but the difference is between that energy transfer of conversion involves the same kind of atoms, like two similar kind of atom transfers energy in between them, and then of conversion process takes place. While in sensitized energy transfer of conversion, to different kinds of atom transfers energy between them. Similarly, there is cross relaxation in which the excitation of one atom leads to the excitation of second molecule and gives luminescence. So this is the phosphor synthesis method. So we have applied hydrothermal synthesis method, in which firstly we prepared rare earth nitrate by dissolving rare earths like polonium oxide and yttrium oxide into the HNO3 solution. The concentration was kept 0.1 molar, and along with this nitrate formation, we prepared trisodium citrate solution and sodium and fluoride solution with water. Then we mix it together to form a gel-like solution. Then we kept it under a high temperature, approx 180 degrees Celsius for 24 hours in a sealed autoclave. Then after 24 hours, we cooled it down to the room temperature naturally, and then centrifugation at 8,000 RPM. By centrifugation at 8,000 RPM, we centrifuged it, and drying for 12 hours gives the prepared, give the desired sample. So we performed XRD spectra to confirm the desired phase of this prepared phosphors. So we got an orthorhombic phase of HOIV code of YF3 phosphor, and by using Devasseur equation, we have calculated the average crystallite size was 0.58 nanometer. So here is the XRD spectra in which the assigned peak or assigned peaks are assigned to the corresponding planes. So now the upcoming emission study. So here this graph shows the variation of intensity of this HOIV code of phosphor with respect to the concentration. Firstly, 1%. We have prepared the different concentration of HO by fixing the concentration of YB concentration YB and concentration was fixed at 10%, and then HO concentration was varied by 1%, 2%, 3%, and 5%. Then this was the variation in emission intensity. So and this is the pump power dependence. So here we can see that the 1% HO is the maximum, giving maximum emission, and then we kept this 1% HO doped YF3 phosphor, and then we vary the power of the laser source. So here we use the laser source of 180 nanometer lasers to excite the sample, and we can see here that there are three emission peaks at 542.5 nanometer, 650 nanometer, and 742.5 nanometer wavelength. Then we have drawn the energy level diagram to see the where transition takes place. So here we can see that the 542.5 nanometer transition, 542.5 nanometer emission is coming due to transition from 5F4 to 5I8 state of HO3 plus ion, and 650 nanometer is coming due to 5F5 to 5I8 nanometer 5I8 energy level, and then 742.5 nanometer wavelength is coming due to the transition from 5F4 to 5I7 energy level of the HO3 plus ion. Here YB absorbs the 980 nanometer photons, and then it transfers to the HO ion, holonium ion. Then the holonium makes transition and gives emission. So here the relation, here the log pump power versus log of intensity relation gives the number of photons involved in this process. Here we can see that the number of photon is more than one, so we can assume it as we can take it as 2. So here it is a 2 photon process. This is down converter photon studies. Firstly, we used 397 nanometer excitation to see the emission peaks corresponding to that excitation spectra, excitation wavelength. So we got two emission peaks, one around 450 nanometer and one around 542 nanometer. So again, we to cross check, we used 542, we fixed 542 nanometer emission to see what excitation can take, what excitation wavelength can be obtained. So we got approximately 397 nanometer excitation. So this is the conclusion that HOIV phosphor has been prepared by hydrogen synthesis method. Then XRD spectra confirmed its phase and average gas flash has calculated at 0.58 nanometer. Then up and down thermal photon spectra has recorded to understand the energy transfer mechanism and number of photons taking part in process. Intense green and less intense red emission has been obtained with the illumination of 90 nanometer laser, diode laser, and then also down conversion photoluminescence study has been performed. So these are some references. Thank you.
Thank you for your presentation.
Yes.
Now we move to the next presentation. OP23 Subham Kumar. Next presentation, OP23. You can start, sir. You can start, sir. Mr. Shubham, you can start your presentation. You can start your presentation.
I think sir mic is not working. Subham, why mic is not working?
His mic is not working.
Yeah.
Hello, am I audible now?
No.
Okay. Good evening, everyone. I am Shubham Kumar from Amity Center for Spintonics Material, Amity University. I would like to thank Sarda University for giving me the opportunity. Now, my topic of the talk is control synthesis of L10 order iron platinum alloy films and to investigate their structural and magnetic investigation. This is my plan of talk. This is my slide is not sharing.
Make it full screen.
Okay.
Slideshow. Yeah.
Okay. So here is my plan of my talk. Now I want, here is the introduction. Since the first commercialized drive was invented, the areal density has been increased 9 of orders. As the digital information is getting doubled in every 2 years, so increasing the data storage capacity is never-ending demand that magnetic recording industry is facing. Now, storing such huge data on multiple servers gave momentum to research, and now there's the quest is for now new hard disk drive with recording densities about 1 terabyte per inch scale. In this viewpoint, the perpendicular magnetic recording materials are high potential candidate for ultra high density data storage media and are believed to be current and future of hard disk drive in various recording methods. So for the potential candidates, iron plate FEPT and COPT emerges as strong potential candidates. Now, the challenge for increasing the storage capacity, writability of the media, thermal stability of the store bit, sufficient number of grains to maintain signal to noise ratio. To increase the storage capacity, we have to reduce reduce the bit size up to few nanometers, and for this we need high anisotropic materials. So thermal stability is one of the key issues, and with high magnetic anisotropic material, the writability issues come into the picture, where because the switching field we need for information exceeds few Tesla. Now these are three conditions: writability, small grain size, and thermal stability, these met simultaneously at a point, and this is called magnetic recording trilemma. So we have to overcome all these three challenges. Now we find a potential candidate, FEPT. The L10 order FEPT has considered the most promising candidate due to its exciting properties, extremely high magnetic anisotropy of order 10⁻⁷, good saturation magnetization, and stability against oxidation. Now I would put your attention towards FCC and FCT structures of Fe and PT. This is FEPT. This is in left side here is FEPT disordered phase. This is FCC structure when Fe and PT atoms are on any random site. But here in FCT, right hand side FCT, no Fe and PT follow a pattern not on random sites. And this can be achieved by chemical processes, ion iridation, and isochronical annealing. This is the phase diagram where we can see on x-axis there is a concentration of PT. So we can conclude that the concentration from 40 to 60% of PT leads to L10 phase. And beyond this, there are different phases not of our interest. Now we move to our sample preparation. The sample are prepared in Amethi Center for Spintronics Metal. This is the instrument, and the sample are prepared by using technique ion beam sputtering, where the basic phenomena of this technique is that the high energy ions fall on target with the help of electric potential, and they knock out the atoms and they deposit on the substrate. Here are some specifications. The Kaufman type ion source produce an ion beam of desired gas, argon, nitrogen, oxygen. We specially use argon gas. Now the beam diameter is 10 mm at source, and energy is 600 electron volt. Now we have deposited three samples, say S1, S2, and S3. Here these are circular Fe targets, and the dotted, we say this is the beam spot. To make different composition of the iron and platinum, we put a platinum foil on iron target, covering different distances of the beam spot. Say in S1, it's below 2 mm from center, and in S2, it's in center. In S3, it's above 2 mm, so that we can get different composition in FEPT. Now, number of deposited iron and platinum atoms will depend on the area of the target exposed to the beam, the deposition rate of iron and platinum, and deposition time. Now, this is here the data analysis. First, the X-ray fluorescence data, in which we take standard FEPT with 50-50 composition and compared with these three samples at S1, S2, and S3. Comparing the intensities and putting in this formula, we got the composition A and 1 minus A shows the relative atomic percentage of Fe and PT. Now, after calculating the value A and 1 minus A, we got S1 and S2 and S1, S2, S3 composition like this, where S3 is nearly equiatomic. Here, this is the X-ray reflectivity data. X-ray technique is used to determine density, contrast, film thicknesses, surface to interface roughness above the critical angle where the rays penetrates into the sample is critical angle is known as the critical angle where the rays pass into the sample and reflected. And this is the experimental data we got for all the samples S1, S2, and S3. And we can see from this formula that the critical angle directly depends upon the densities. And the sample S3, which has less dense, is have less critical angle, and the thickness found is 850 angstrom for all the films. Now here is the X-ray diffraction data. On left hand side, there is as deposit data, and on right hand side annual data. We can compare them, and we found there for all the samples there are different peaks which we got after the annealing the sample, say 001, and the splitting here the 002 peak, and this shows that there is another phase and confirms the L10 phase. This is a mock data, and to investigate the magnetic properties, the mock data is taken by applying the magnetic field, and for all the samples, S1 and S2 and S3, S1 where the platinum concentration is highest, and S2, S3 is nearly equiatomic. So we can see the variation in the S1 is shows non-linear magnetization, and S2 and S3 we can say is linear, shows linear magnetization. And also confirms L10 phase of our requirement. Now the conclusion that the nearly equiatomic composition of iron and platinum, which is crucial for its practical application, is optimized and successfully achieved using iron beam pattern. Composition of the films confirmed from X-ray fluorescence and X-ray reflectivity measurements. L10 ordering in optimized FPT film is observed via XRD measurements, and magnetic properties were examined using mock measurements. So here I would like to acknowledge Dr. Kavita Sharma, Dr. Gagan Sharma, and rest of my faculties and our department, Amity Center for Spintronic Metals and funding agency, UGCDA. Thank you.
Thank you for your presentation, Shubham Kumar. Yeah, now we will move to next presentation, OP24, Shivchandra. Yeah, next presentation, OP24, Shivchandra.
Yes, sir, I'm sharing, sir.
Yeah, sure.
Can I start, sir?
Yeah, you can start by using slideshow. Just click on slideshow at the top. Yeah. After animation, slideshow is there at the top. Next. It's the right side. Next, yeah, from beginning. Yeah, you can start.
Good evening, everyone. I'm Shivchandra, PhD scholar from IIT Madras. Today I'm going to present the oral presentation, biogenic synthesis of zinc oxide nanoparticles using Annona muricata plant leaf extract and its anti-cancer efficacy on 2D and 3D tumor models. This presentation contains nanoparticles in drug delivery, green synthesis of nanoparticles, phytochemicals characterization of Annona muricata zinc oxide nanoparticles, and AMZN1 nanoparticles for biomedical application, anti-tumor effect on 2D and 3D cancer models, mechanism of cancer cell death, and acknowledgement. Nanoparticles. There are different types of nanoparticles. Those are lipid-based nanoparticles, drug conjugates, and polymer-based nanoparticles, and viral nanoparticles, and inorganic nanoparticles. My area of interest is metal nanoparticles. Actually, metal nanoparticles synthesis can be done by physical and chemical synthesis methods. This requires excess of organic solvents and toxic chemicals, and this causes harm to the environment. To address this problem, we synthesize zinc oxide nanoparticles using plant leaf extract, which contains phytocompounds. and it involves in the cancer treatment and prevention. Here, green synthesis has different types of applications. One is it is eco-friendly. You can synthesize very large productions. Second one is third one is we need no need of energy saving process. Here, synthesis of the zinc oxide nanoparticles. We extracted the leaf compounds by boiling plant leaves, and we squeezed it, and after that we added in the zinc acetate solution in 50 ml of water. Then we added into 2 molar NaOH to make the solution pH 12. After that we separate. White precipitate is formed. It indicates that the formation of zinc oxide nanoparticles. After forming the zinc oxide nanoparticles, we done that the physiochemical characterizations to know that different functional group present in that compound. For that, we did FTIR to know that the functional group present in that compound. In this FTIR, 3470 and 3405 indicates that OH stressing, and 1639 indicates that alkane group, and 15163 indicates that aromatic ring. And these compounds includes amines, alcohols, alkanes, carboxylic acid group, suggests that the compound contain, actually phytocompounds contain these all functional groups. And it is used for hydrolyzing agent of zinc oxide nanoparticles. After that, we did UV visible spectral analysis. We found that peak 355 nanometers, which indicates that zinc oxide nanoparticles. And after that, we did X-ray diffraction analysis for purity and crystalline nature. Here we didn't found any peak respective to zinc acetate, which indicates that formed compound is very pure, and we found that average crystalline size is 16.79 by using that Debye-Squarcher equation. After we did TEM analysis to know the size and shape of that nanoparticles, we found that the nanoparticles in a spherical shape, and after that we did particle size is around 80 nanometers. The DLS and zeta potential, we did hydrodynamic size, and the particle distribution is 417 nanometers, and the particle distribution is 0.886, and average zeta potential is minus 2.59 MV. This indicates that it doesn't have a reflective force. This means the nanoparticles will agglomerate. To overcome this problem, we are using the sonication to uniformly distribute that zinc oxide nanoparticles. After that, we did the biocompatibility test on 3T3 cell lines. It showed that 80% of cells are viable. So only 20% is toxicity to the cells. After that, we did hemocompatibility test on 2% RBC. In this, with the different concentrations, 10, 5, 2.5, and 1.25, and negative control and positive control. Negative control is PPS, and positive control is water. So we found less than 5% of hemolysis occurred, which indicates that we can use this as a zinc oxide. We can use in the biomedical applications. That means we can inject in the intravenous applications. If more than 5, we cannot use it for intravenous applications. So these zinc oxide nanoparticles are biocompatible and hemocompatible. So after that, we did anti-cancer efficacy experiments. Miyapaka cell lines, those are pancreatic cells, and mold core cells, those are lymphoblastic leukemia cells. In Miyapaka cells, we did we found that IC50 values are 96.67%, and in mold for cells, it is 60.13 micrograms per ml. Next, we did 3D tumor model, that is spheroid model. It represents that tumor microenvironment. We used A549 cell lines, lung cancer cell lines. We used different concentration, that is IC50 and of IC50 values, and 3 different time periods. One is 24 hours and 48 hours and 72 hours. We use doxorubicin as a positive control, and we found that diameter of the spheroids are reducing due to depletion of the nutrients in the media. Then we did mechanism of cancer cell apoptosis assay. In apoptosis assay, there are four different phases. In one, Q2 is the late apoptosis. In this, we found more number of cells are dying. That is, in of IC50 values, the percentage is 29.9%, and IC50 values it is 59.5%. It indicates that more number of cells are dying in late apoptosis. We did mitochondrial membrane potential assay for IC50 and half IC50 of zinc oxide nanoparticles. So the membrane potential change is increasing as the concentration of that nanoparticles increases. Future study, we want to do in vivo study using a different concentration, that is 25 mg, 50 mg, and 100 mg per kg body weight. Each has 3 controls, and after the 11th day, you have to euthanize this thing, and you'll use it for histological assay and hematological analysis and biochemical analysis. Coming to conclusion, we synthesize that zinc oxide nanoparticles using unknown muricator plant, and the synthesized nanoparticles are biocompatible and hemocompatible in nature. And we did anti-cancer effect studies on 2D model and 3D model. These nanoparticles are effective in cancer cell death. Collaborators, thanks to collaborators for supporting this work. Thank you.
Hello, any question from audience? You can unmute and directly ask the questions from Save. Okay, there is one question in Dropbox. Can you explain the reason of choosing docs in comparison with vivo study with your nanoparticle? Shiv?
Actually, earlier studies, it showed that it is very effective in cancer treatment, sir. So for that reason, we are using that doxorubicin as a control, sir. Hello?
Yes, I'm hearing you. Nautlina Salim, you can unmute yourself and directly ask the questions to Shiv Chandra. Anji sir? Just 1 minute. Sir, now Neha Bansal is available. Sir, OP25. Yes, sir, you can unmute, sir.
Yeah. Next, we will move to next presentation, OP25 Neha. Next presentation, we will move to OP25 Neha. Neha is there?
Yes, sir. Neha is in the meeting.
OP25 Neha.
Neha Bansal, please respond.
Neha Bansal, OP25. Yeah. Now we move to OP20.
Yes, sir. Move to the next.
Sir, I'm Neha Bansal.
Okay.
Am I audible? Am I audible?
Yeah, audible.
Yes, sir. Sir, actually, I'm not looking at the. I'm looking at the option where I can set the virtual image, but
No problem. You can share
that is
your
fine.
screen.
I can start with my presentation.
Yeah, you can start. You can share your screen.
Okay. Just a minute, sir. So I will share my screen for the old presentation. Sir, my presentation is visible to you?
Yeah, visible. Yeah, you can start.
Okay. So good evening, everyone. I'm Neha Bansal, PhD Research Scholar in EEE Department in School of Engineering and Technology, Sharda University. My PhD research is on long-term performance assessment and reliability assessment of solar PV plants installed in India. And today my oral presentation is on long-term performance assessment and energy loss analysis of 9 megawatt grid-connected solar photovoltaic plant in hot and dry climate in India. The outline of my presentation consists of introduction, description of utility scale 9 megawatt PV plant, methodology adopted, results and discussion, followed by conclusion. Consists of comprehensive performance investigation of 9 megawatt grid connected utility scale solar photovoltaic plant. Over here, megawatt peak and above, and the plant which I have studied is of 9 megawatt capacity. Now this plant is installed with 3 different PV technologies, that is polycrystalline silicon, amorphous silicon and cadmium telluride in hot and dry climate in Kutch district of Gujarat, India. Now, this hot and dry climate of Kutch district is adopted from the widely used climate classification system in India by Bansal and Minke, who studied the Indian climate in detail and divided the Indian climate into 6 climatic zones: hot and dry, hot and humid, temperate, cold and sunny, cold and cloudy, and composite. Now, this hot and dry climate is characterized by monthly mean temperature of greater than 20 degrees Celsius, the precipitation less than 5 mm, and the number of clear sunny days in a month greater than 20, and the relative humidity less than 55%. Now, the performance of any solar PV plant essentially depends upon 2 factors. First is the environmental parameters of the plant location, and second is the type of PV technology used. Now, over here, the environmental parameters of the plant location consist of the available incident solar irradiation, ambient temperature, wind speed and direction, humidity, precipitation, and soiling. Now, to analyze or to evaluate the performance of any solar PV plant, there is one standard documentation, which is the International Electrotechnical Commission documentation IC61724, which determines the solar photovoltaic performance monitoring guidelines for measurement, data exchange, and analysis. Now, as per this standard documentation, to evaluate the performance of any solar PV plant, various performance indices need to be evaluated. These performance indices are reference yield, area yield, final yield, performance ratio, capacity utilization factor, area capture and system losses, PV array, and system efficiency. So all these indices I have evaluated based on 4 years of actual plant monitoring data and PVC simulation data for 25 consecutive years. The main objective of the conducted research is divided into 3 sections. The first section consists of to evaluate the performance of plant with increase in outdoor exposure period. Second is to carry out error analysis of the simulated and actual plant generation data. And third is assessing the suitability of type of PV technology in hot semi-arid climate based on comparative performance assessment of crystalline and thin film PV technologies. Now, this is the actual site view of the 9 megawatt solar PV plant, and these are the installed polycrystalline silicon, amorphous silicon, and cadmium telluride modules. Now, there are total 46,361 PV modules, out of which 36,363 modules are of polycrystalline silicon technology of 230 watt peak, constituting 8 megawatt. So basically, now the plant capacity is 9 megawatt, out of which 8 megawatt consists of polycrystalline silicon technology, and the remaining 1 megawatt is attributed to thin film PV technology, where 0.5 megawatt is consists of amorphous silicon technology, and 0.5 megawatt consists of cadmium telluride technology. There are total 18 central 500 kilowatt inverters connected in the plant, And there are total 9 1 MVA transformers for stepping up the voltage from 450 volt to 11 kV, and one main transformer of 12.5 MVA capacity for stepping up the voltage from 11 kV to 66 kV to be fed into grid HT lines. Now, these are some technical specifications, some key parameters of the PV modules that are rated at STC. Now, these are the electrical parameters of the module. Two important parameters to be noted here is the module efficiency. So, the module efficiency of the polycrystalline silicon modules installed is 14%, of amorphous silicon is 6.4%, and of cadmium telluride is 14.6%, whereas the temperature coefficient of maximum power is highest of polycrystalline silicon technology, that is minus 0.43%. Over here, the temperature coefficient of maximum power means with every 1 degree Celsius rise in ambient temperature, the corresponding decrease in the module output power is by minus 0.43%, and the least temperature coefficient of power corresponds to amorphous silicon technology with minus 0.25%. Now, methodology. Now, the PV system, just a minute. The methodology is divided into 4 sections. So first, the first section consists of PVC simulation of the plant for 25 consecutive years. Second is evaluation of plant performance indices as per IC 61724 based on actual plant monitored and simulated data. Third is comparison and error analysis between simulated results and actual monitored plant data. And fourth is comparative performance analysis of polycrystalline silicon, amorphous silicon, and cadmium telluride PV technologies, and evaluating suitability of PV technology in hot semi-arid climate based on the results obtained. So in the results and discussion section, the first this section is also divided into 4 subsections. The first section consists of performance analysis based on actual plant monitoring data. So this figure represents the plane of array irradiation received and the ambient temperature profile of the plant location for the entire monitoring period of 4 years from 2012 till 2015, month wise from January till December. So the climate of the plant location is divided into 4 seasons. Now, the summer season consists of 4 months, that is March, April, May, and June. So we can see that the ambient temperature is more than 30 degrees Celsius in the month of April, May, and June. Then it significantly starts decreasing in the rainy season, which comprises of July, August, and mid of September. Now, after that mid of September, we can see a general increase in the solar radiation intensities and the ambient temperature. So this is the post-monsoon period, which consists of mid of September till October, and then the winter season comes, which consists of November, December, January, and February, wherein the ambient temperature significantly decreases to 18 to 20 degrees Celsius, and the irradiation intensity, but the irradiation intensity is maintained well above 6 kilowatt-hour per meter square per day. Now, this graph represents the reference yield and the final yield of the plant in hours per day. Now, over here, the reference yield is the number of peak sun hours in a day, and final yield determines the number of hours in a day during which the plant operates at its rated power. So we can see that there is considerable difference between the reference yield and final yield, and this is attributed to high capture losses in the plant. Here, the capture losses consist of the optical losses, temperature losses, soiling losses, shading losses, the DC wiring losses, and the degradation losses that are being observed in the fielded PV modules. So from this figure, we can see that there is linear decrease in these values over the with increase in outer exposure period. That is from 2012 to 2015. We can see that there is linear decrease in the plant final yield. Now, the next figure represents the performance ratio of the plant and the capacity utilization factor of the plant. So performance ratio of the plant essentially determines the actual performance of the plant with respect to the ideal performance, and the capacity utilization factor determines the actual energy generation of the plant with respect to the generation if the plant is operated for 24 hours in a day and 365 days in a year. Now, over here, we can see that this PR value, the starting PR value is very low in March 2012. And this is attributed to the high light-induced degradation losses. We know that the plant is of 9 megawatt capacity, and predominantly the 8 megawatt capacity is occupied by polycrystalline silicon technology. Now, in case of polycrystalline silicon technology, the initial light-induced degradation losses account for initial 1000 hours of sunlight exposure. So these 1000 hours, if we look into days, then it contributes to around 41 days. So we can see that initially for this March 2012, the PR value value is less, owing to less energy generation of the plant, and thereafter the PR value rises. Now we can see that there is this kind of a dip can be seen everywhere in all 4 years, in all 4 consecutive years, and this corresponds to the rainy season, because in rainy season very less irradiation intensities are available, and therefore less generation is there and less PR value is obtained. And we can see that PR values they increase linearly in the winter months, and they reach peak value in the summer months, particularly in March and April. Now this figure represents the array efficiency and the system efficiency of the plant. Now to understand this particular array and system efficiency, we need to understand the capture losses and system losses of the plant. So basically the capture losses are the losses that occur on the DC side of the plant, and the system losses are the losses that occur on the AC side of the plant. And these system losses are nothing but the inverter operation losses, the transformer losses, and the AC wiring losses, which are very minimal, and they contribute to around only 0.09 hours per day, and they remain constant, which can be seen that these values have remained constant over the entire monitoring period. There is the we can see that there is linear increase in the capture losses of the plant, and because of this linear increase in the capture values of the plant, the array efficiency and the system efficiency of the plant has decreased over the entire monitoring period. Now, this was all about the performance evaluation of the plant based on actual plant monitoring data. Now, the second section of the plant consists of performance analysis based on PVC simulation data. So now if we want to predict the performance of the plant, and if you want to understand the performance of the plant in hot semi-arid climate, how it is going to perform, then PVC simulation tool has been used in evaluating the plant performance for 25 consecutive years. So the first figure represents the energy. EAC is basically the energy fed into the grid, and this represents the first year of operation, 5th year of operation, 10th, 15th, 20th, and 25th year of operation. So we can see that there is linear decrease in the annual energy generation of the plant, and this matches well with the actual monitored values also. Now we can see the next graph represents performance ratio and capacity utilization factor. Now, since this capacity utilization factor essentially depends upon the annual energy generation, we can see the linear decrease in capacity utilization factor values, starting from 19% in the first year to 13.6% decrement in the 25th year of operation. And similarly, in case of performance ratio, the performance ratio in the first year of operation is observed to be 74%, and it gets reduced to 53% in the 25th year of operation due to plant degradation. Now, the next comes is the capture and the system losses based on simulated data. And this is the PV array and system efficiency based on the simulated data. So we can see for entire project service lifetime of 25 years, there is a linear increase in capture losses from 1.46 hours per day in the first year to 2.9 hours per day in the 25th year. So as the capture losses are increasing linearly, the linear decrease can be seen in the PV array and the system efficiency. And I have already mentioned earlier that this PV array and PV system efficiency, there is a marginal difference between the PV array and the system efficiency, because the losses on the AC side are very less, and the losses primarily are on the DC side. And that is why there is a huge difference between the reference and the final yield, but the difference between the array and final yield is minimal. Now, third is the error.
Please conclude.
Hello.
Please conclude.
Sir, error analysis, I need to explain. This is the error analysis.
Almost more than 10 minutes.
Only 1 minute, sir, please. 1 minute, sir. Sir, this is the error analysis which I have performed, and you can see this black dots correspond to the simulated values, and these are the these blue dots correspond to the actually measured values. And we can see there is very good agreement between these two values, and the total annual error analysis indicates 7% of error between the measured and the simulated values. And these are the comparison of various polycrystalline cadmium telluride and amorphous silicon technology in first year and 25th year of operation. And with respect to the energy generation and PR, we can say that the optimum technology in this hot semi-arid climate will be cadmium telluride technology, followed by polycrystalline silicon, and in last by amorphous silicon. And this is also this annual energy density is a very important parameter to be noted, because it is basically the energy generation from the plant with respect to the area occupied by the PV technology. So we can say the highest area, that is the highest area is occupied by the amorphous silicon technology. So in urban places or in places where land is a constraint, one should go for cadmium telluride technology and polycrystalline silicon technology. So the conclusion of my presentation consists of the annual energy generation of the plant, the final yield of the plant, which has seen to decrease from around 5 hours per day to 3.28 hours per day. The performance ratio has reduced from 74% to 53% in 25th year. Similar reduction can be seen in capacity utilization factor and system efficiency values, which are around, which have decreased from 11% to 7% in 25th year of operation. The annual temperature losses are observed to be around 14.9%, contributing to 2970 megawatt hour per year. And the mismatch losses have ranged from 2 to 7%, contributing to 350 to 880 units of loss annually. So based on the comparative performance analysis, we can say that the optimum PV technology in hot semi-arid climate of Kutch district, Gujarat, India is CDT, and the second technology which the PV plant owners must consider should be of polycrystalline silicon technology. So the findings of this research work, of the conducted research work, will be useful to EPC contractors and PV plant owners in understanding long-term performance of solar PV plant in hot semi-arid climate, and will help in better design engineering of future solar PV plants. So thank you, everyone. I hereby conclude my presentation. I'll be glad if, in case there are any queries, I will be glad to answer them.
Thank you for your presentation.
Yes.
You can message in chat box. Next, we will move to the next presentation, OP26, Komal Gupta.
Hello.
The next presentation, OP26, Komal Gupta.
Yes, sir. Am I audible?
Yeah, audible.
And my screen is visible to you?
Visible, but slideshow. You can keep it in slideshow.
Yeah, sure. Now is it okay?
Yes, okay. You can start.
Yeah. Hello, everyone. I am Dr. Komal Gupta. I am assistant professor in the Department of Chemistry, Multani Mal Modi College, Modinagar, Ghaziabad. Today, I am going to present my research work on the plant extract mediated zinc oxide nanoparticles, synthesis and application. So first of all, I would like to give a brief introduction about the nanotechnology. It involves the manipulation of matters on an atomic and molecular level, and it deals with the developing material, devices, or other structures possessing at least one of its dimension size from 1 to 100 nanometer. Nanomaterials has been classified in different categories such as nanoceramics, metal nanoparticles, carbonaceous nanomaterials, and semiconductors. Different semiconductor nanomaterials includes the pure elements of fourth group, binary compounds, different compounds of third, fifth group, second, sixth group, and the fourth, sixth group of periodic table, different oxides, alloys, ternary compounds, and composites. Among them, second, sixth semiconductor nanostructures are the different chalcogenides of zinc, cadmium, and mercury. Among them, zinc oxide possesses different properties, such as it is functional, strategic, promising, and versatile inorganic material with a broad range of applications. It is characterized by a direct band gap of 3.3 electron volt and near UV spectrum with a high excitonic binding energy of 60 electron volt at room temperature and a natural n-type electrical conductivity. The wide band gap of zinc oxide has a significant effect on its properties such as electrical conductivity and optical absorption. It shows light covalent character, very high, very strong ionic bonding of zinc bond oxygen. Its longer durability, higher selectivity, and heat resistance are preceded than other organic and inorganic materials. There are different methods for its synthesis, which are physical methods, chemical methods, and biological methods. Among different methods, most important are the biological synthesis methods, which are comparatively greener than other methods. And biological synthesis methods includes their synthesis using waste material, using microbes mediated synthesis such as algae, fungus, bacteria, and the other one is the plant mediated synthesis. So in our research work, we have used plant extract as a template for the synthesis of this zinc oxide nanomaterial, which is because that plants are the nature's chemical factories. They are cost efficient and require low maintenance. Plant-based synthesis of nanoparticles is faster, safer, and lighter, works at low temperature, and requires only modest and environmentally safer components. Plant-based nanoparticles have attracted more attention due to growing interest in environmentally conscious products. The synthesis of nanoparticles using plants offer other advantages, such as the utilization of safer solvents, decreased use of dangerous reagents, milder response conditions, feasibility, and their adaptability in use of medicinal, surgical, and pharmaceutical application. So in our research work, we have used Beccalia nilotica plant, which is also known as babool, leaf extract for the synthesis of zinc oxide nanomaterial, which is due to its importance. It is Beccalia nilotica is known as a indispensable medicinal properties. It is majorly used for cleaning of the teeth, and the regular use for its facilitates its strengthening the gum, teeth, and reduce the plaque and inflammation. The leaves of babool are invaluable in healing the wounds. Leaves and bark of the babool possess potent and anti-inflammatory and antibacterial properties which aid in controlling bleeding and infections, which speeds up the healing wounds, cuts, and injuries. The powerful anti-inflammatory, antiseptic, and antifungal properties of babool leaves are beneficial in treating various skin type conditions, and different phytochemicals present in this in the leaf extract, like flavonoids, phenols, alkaloids, all makes it as a template for the nanoparticle synthesis. So this is the methodology for the synthesis of zinc oxide nanoparticle. We have used Soxhlet apparatus for the leaf extraction, and this leaf extract and was added in zinc acetate solution and was stirred for 2 hours. And after that, we have maintained its pH at around 12 by using sodium hydroxide solution. And then it was stirred for 2 hours, and then it was purified by redispersion in deionized water and then centrifuged, and then it was dried overnight in the oven at 60 degrees centigrade. The synthesized nanoparticles were characterized using different techniques. Their structural identification was carried out using XRD. Their morphology and composition was studied by FESM in EDS analysis. Structural interactions was carried out using FTIR spectroscopy, and their optical studies was carried out by spectrophotometer and spectrofluorophotometer. So these are the optical study of the plant-mediated zinc oxide nanostructures. The absorption spectra of these nanostructures shows an absorption peak at 358 nanometer, which is actually blue shifted in comparison to the bulk zinc oxide nanoparticles. And the excitation of these nanoparticles with the 340 nanometer excitation wavelength shows 2 emission peaks at 380 nanometer and 469 nanometer. The peak at 380 nanometers is supposed due to the near band gap excitonic emission, and the peak at 469 nanometer correspond to the presence of singly ionized oxygen vacancies. The XRD analysis shows different planes at different 2 theta angles, which confirms its phase to be hexagonal Woodside phase of zinc oxide, and it perfectly matches with the JCPDS number 361451, and it also shows formation of the nanocrystalline nature of zinc oxide nanostructures. These are the FESM analysis images. The FESM images shows the formation of spherical nanoparticles, and we have also calculated the nanoparticle size using images software, and the average particle size has been calculated to be 30 nanometer with a particle size distribution from 10 to 50 nanometer. We have also did the EDS analysis, which confirms the presence of different components in the zinc oxide nanomaterials sample. EDS mapping also suggests the presence of zinc, oxygen, carbon, and nitrogen, which are due to the phytochemicals present in the leaf extract. The FTIR analysis shows different peaks corresponds to the moieties, different moieties present in the phytochemicals such as alkanoids, phenols, etc., and also due to the zinc oxides. And we can see that in the presence of plant extract, the peaks due to different zinc oxide get shifted, which shows its interaction with the plant extract. So we know that the zinc oxide has many applications in the field of photocatalysis, textile industries, electronics, biosensing, and other biomedical applications. The zinc oxide nanoparticles has been used as a new antimicrobial agent. The integration of nano-sized zinc oxide with biocompatible Vaccalia nilotica leaf extracts will make it more biocompatible and more efficient antimicrobial agent, which can be used in different biomedical fields and also in the cosmetic industries. And zinc oxide possesses high optical absorption and in the range of 315 to 400 nanometer and UVV region from 282 to 315 nanometer region, which is beneficial in antibacterial responses and used as a UV protector in cosmetics. Jatano has also found high photocatalytic efficiency among different inorganic photocatalytic material. Photocatalytic degradation oxidizes hazardous complex organic compounds like organic dyes such as methylene blue, methyl orange, rhodamine B, etc. Increased surface to volume ratio due to decreased particle size in the synthesis will enhance the photocatalytic efficiency for the synthesized nanoparticles. So these are the outcomes of my research. Use of Vaccalia nilotica leaf extract as a template for the synthesis of zinc oxide makes it greener, biocompatible, and sustainable nanomaterial. Different phytochemicals present in the leaf extracts like flavonoids, alkanoids, phenols, it stabilizes the zinc oxide nanoparticles with different functional group interactions. Synthesized zinc oxide shows blue absorbance compared to bulk form, suggesting the decrease in size on the integration with phytochemical. Fluorescence from the particles makes it nanoparticles. The FSM analysis 10 to 15 nanometer. FTA analysis shows its interaction with different functional groups present in the phytochemicals and the synthesis of
It was disconnected.
Hello, sir, there is some connection issue, I think.
Yeah. I think Komal Gupta have completed your presentation. You can unmute. You can unmute yourself.
So just 1 minute.
Hello.
Yeah, have you completed your presentation?
Yes, sir. It's almost completed, but I lost the connection in between.
Okay, it's no problem.
Yeah, so just I would like to acknowledge ICSEM team for giving me this opportunity. I would also like to acknowledge my principal Nagar and also to IIT Roorkee for helping me out in the different characterization processes, and to the head of the chemistry and physics and botany department Multani Mal Modi College Modi Nagar for their kind help. Thank you so much.
Yeah, thank you for your presentation, Komal Gupta. Yeah, now we move to next presentation, OP27, Vasudha Sharma. Yeah, OP27, Vasudha Sharma. You can share your screen, Vasudha Sharma. OP27. Vasudha Sharma, you can share your screen. Vasudha Sharma.
Sir, I think mic is not working. Vasudha sir, mic is not working, sir.
Yeah, I think our mic is not working.
Wait for 1 minute.
I think this is the last presentation in this session.
Yes, sir. Yes, sir. This is the last presentation. Yes, sir. No. We are sharing this.
But your voice is not from Dr. Vasudha Sharma. Check with your mic. No audio.
Sir, there is a problem in mic, sir. Yes, sir. Wait for one.
Screen is sharing, but no audio.
Yes, sir. Mic is not working, sir, because there is no fluctuation in mic, in audio. Ma'am, please join the meeting with the mobile phone.
Mic is not working. Try to connect to the mobile.
Oh, so ma'am, please connect from the other device. There is some problem in mic in your system.
Is it possible to change the device? Just we can message in
So I have already informed.
message in chat. That's one. There is possibility to change the device.
I think so there is the same problem with the mic.
She asked 1 minute.
Yeah. Here I have given the full rights. They are the co-host of the meeting. Yeah, they have joined with a different device.
Hello. Good evening, sir.
Good evening.
I'm so sorry. I do not know why this trouble came up.
You can start.
Yes, thank you, sir. I thank you all for having some patience with me. Very good evening to all of you. I'm here to present my work, that is green synthesis of zinc oxide nanoparticles using plant extract as a template. So
please make it full screen. Slideshow. Yeah, first from the.
So nanotechnology has been recognized as a new science of 21st century and has a role in chemical sciences, material sciences, and physical sciences, biomedical sciences, and engineering. Due to nanotechnology, different properties of matter, that is optical, magnetic, and electrical, can be controlled. Nanomaterials can be formed. These materials are single unit, small sized material ranging between 1 and 100 nanometers. At nanoscale level, alterations in size and shape occurs due to increased surface volume ratio, which affects the physical properties of materials. Among the nanomaterials, metals and metal oxides have shown the highest potential. Oxides of transition metals have found application in solar energy, processing, magnetic, solar storage, electric catalysis, and role as a microbiocide. Nanomaterials can be formed by different methods. However, green nanotechnology has emerged as eco-friendly, cost-effective method. Green nanotechnology reduces the need of harmful chemicals, and different biomolecules can be used as reducing or capping agents. Green nanotechnology also eliminates toxic elements that may have been left out in the process and in trace concentration, and they may hamper the final application of the nanomaterial. The crop that I used as my plant material was sunflower. So sunflower is a member of the family Asteraceae and is an economically important crop worldwide. The inflorescence, which is also known as the capitulum or the heads, contain seeds at maturity. All the parts of the plants have economic importance. The inflorescence of the sunflower are used for ornamental process purposes. The pollen is collected by the honeybees for the formation of nectar. The seeds of sunflower are eaten in raw or roasted form and are also used for the formation of the oilseed. The leaves and stem are used for the fodder purposes. The plant material chosen was sunflower, that is Helianthus annuus variety KBSH. The seed husks are actually the byproduct of the oilseed industry, as the seeds are used for their rich oil contents. The seed coats are a byproduct and are a waste product. The cultivated varieties, in this case KBSH, also lose their viability after some time for about 1 or 2 years. So after 1 or 2 years, they are agriculturally non-viable. So from these waste material, nanoparticles have been created. So as for the synthesis of the zinc nanoparticles, they were created by co-precipitation method, and the seed coats of sunflower thus obtained. They were subjected to Soxhlet treatment in around 80% of methanol. So most of the flavonoids, phenols, and other biomolecules are soluble in methanols. So these biomolecules actually they react with the zinc acetate, and due to their hydroxyl, carboxyl, and amine functional group, they react with the metal ions and reduce them into nano ranges. The biomolecules also help in capping of the nanoparticles, which is important for stability and biocompatibility of the nanomaterials. The UV visible absorption spectroscopy is used to determine the optical property of the nanomaterial thus created. The UV visible absorption pattern shows that an absorption at 360. The purity of zinc nano oxide nanostructures was confirmed by the non-existence of any other absorption peaks. The blue shifted absorption peak is relatively due to the bulk and zinc oxide nanoparticles. Photoluminescence is used for the characterization of optical and electronic properties. Photoluminescence properties show zinc nano-oxide nanopowder has excitation emission of 340 nanometers and two emission peaks of 377 and 469 nanometers attributed to the presence of zinc and oxygen vacancies. The XRD diffraction patterns also confirms the presence of nanoparticle zinc oxide. The diffraction peaks indicates the material consists mainly of the particles in nanoscale range. The results are similar to as that has been reported earlier. FESEM shows the surface morphology of the synthesized product, and it was characterized by the FESEM, and it can be seen clearly that the ZnO nanoparticles, they aggregate to form a prism-like structure. EDAX analysis shows that there are two major peaks of zinc and oxygen between 0 and 2 and two peaks between 8 and 10. These results correlate with the already reported results, which has similar peaks, and they have been observed as zinc nano-oxide nanoparticles. EDAX mapping show the phase information in which the presence of zinc and oxygen is in maximal range along with carbon and nitrogen. FTIR plots the intensity of infrared spectrum and calculates the functional group for the synthesis of zinc nanoparticles. FTIR was scanned from 2000 to around 400 per centimeters in percentage transmitting mode. The zinc oxide nanoparticles show a peak at around 1630, 894, whereas zinc oxide obtained by the plant extract was at 1633, showing the OH stretch peaks, and also it revealed the presence of aromatic rings, aldehyde rings, which were due to the plant extract, and these enhance the stabilization of the zinc nano-oxide particles in aqueous medium. The peaks at 500 and 436 show the stretching of Zn and O nanoparticles. The nanoparticles are highly toxic to a large range of microbial agents. Antimicrobial properties were studied against Staphylococcus aureus and E. coli, that is the gram-positive and the gram-negative bacteria. This diffusion method and microdilution analysis were used to determine the antibacterial efficiency. Zinc oxide nanoparticles interact with the membrane, causing disrupting membrane and causing a loss of integrity of the membrane, causing leakage of ions. Zinc oxide also leads to generation of reactive oxygen species, causing oxidative stress. This causes denaturation of proteins, mitochondria, impaired metabolism, leading to cell death. Hydrogen peroxide has been also correlated as a signaling molecule, and this causes impaired DNA replication, transmission, and also causes damage to the bacterial cell. The institution nearby my college has a lot of textile industries. So textile industry usually give out azo dyes as a waste material. These are harmful for the environment as well. So the photocatalytic activity of the zinc oxide nanoparticles could be seen by the degradation of methyl orange in aqueous media. Initially, a blank experiment was carried out to see the self-decomposition under UV light, and then it was compared to the dye solution with a catalyst. The decoloration of the dye is mainly due to the OH radical and the dye or intermediate photoproducts. The size, morphology, and the shape charge properties of the metal oxide nanoparticles also contribute to the degradation of the color dye. Zinc nanoparticles, they are competent photocatalysts because they produce hydrogen peroxide more efficiently. This increases the surface area of zinc oxide nanoparticles and causes detoxification of the azo dyes, mainly in this case in the methyl orange. So these are the major conclusions of my work. That is, the zinc oxide nanostructures can be created with the help of sunflower husk husk, which is actually a byproduct of the oilseed industry. Green synthesis eliminates the use of toxic chemicals and causes the formation of zinc nanoparticles having various applications. The absorption of zinc oxide nanostructures are blue shifted and have an absorption peak at 360 nanometers. Absence of other peaks reveal the purity of zinc oxide nanoparticle thus created. The photoluminescence shows zinc oxide nanostructures with excitation wavelength of 377 and emission wavelength of 469 nanometers. The EDAX also confirms the presence of ZNO nanoparticles, while FESM shows the formation of prism-like nanoparticles. FTIR reveals the presence of phenolic compounds like amines and carboxylic groups on the surface of nanoparticles, which enhance the stabilization of zinc oxide nanoparticles. Zinc oxide nanoparticle structures, they show antibacterial properties and they also show the photocatalytic activities. I'm thankful. Again, this is my work that I wanted to share with all of you. I'm thankful to all of you for having the patience that I had initially. Thank you.
Thank you for your presentation, Vasudha. We have one question.
Yes, sir.
Can this zinc oxide nanoparticle be an antimicrobial material for other types of microbes such as virus, fungus, or alligator?
Yes, there has been work on the zinc nanoparticles, on the antimicrobial properties of zinc oxide, since they have been created by the chemicals, that is the aldehyde groups and the carboxylic groups and the basically flavonoids which are present in the plant material. So these act as agents for the and cause the disruption in case of the metabolism in fungi and other microbes. In case of viruses, I cannot say this because viruses are a transition. They are among the living and the non-living groups. So maybe not viruses, but they can be used as antimicrobial material for the fungi and the algae also. Though I have not worked on it, but I have seen reports on this matter as well.
Okay. Thank you. Thank you very much.
Thank you so much, sir.
Anji sir, yes sir, now the session is over. All presentations are over from OP1 to OP27. Now from the organizing committee, I must thankful to Anji Polu sir for chairing this long session from 1:15 to till 6:12. This is a very long session, sir. Thank you, sir. And I must thankful to our invited speakers and our participants for continuously in this session one day. So thank you to all, sir. We will meet tomorrow at 9:30.
Okay, thank you very much.
Thank you, sir.